Hi everyone, am I audible to you? Hello? Am I audible to you? Hi students, am I audible to you? So, good evening everyone. Welcome to Unacademy NEET English. I'm your biology educator Ambika Sharma. Welcome to our channel. Good evening. Do let me know, am I audible or not? Yes, am I audible or not? Good evening, good evening, good evening. I hope you all are doing very well and I hope you all are taking a good care of your health as well as your studies. So, good evening people. Good evening. Hi. Voice is low. Is it so? Yes, Girija. I don't know why do some students they complain about the voice, because even after the class when I check the video na, it's perfectly fine. It's perfectly fine. Okay, so welcome to the class. Bache, are you ready to complete one more chapter? Tell me. Are you ready to complete one more chapter? Today we are going to finish biotechnology and its application, and as per your new NCERT, it is your chapter number 10, right? As per your new NCERT. So let's revise it quickly. Let's revise what we have covered so far. So can you just tell me the name of the chapters that we have completed here in this Game of NEET series? Tell me people. Can you just tell me the name of the chapters that we have completed here in this Game of NEET series? Yup, tell me. Let's start from the starting. So we have completed human reproduction, okay, reproductive health, sexual reproduction in flowering plant, yeah, but Pankhudi ma'am taught that, right? So human reproduction, reproductive health, principles of inheritance and variation, molecular basis of inheritance, evolution, right? Evolution. And then biotechnology, principles and processes, and today we'll be finishing biotechnology and its applications. So that is what we have covered so far, isn't it? That is what we have covered so far. And on 23rd of Jan, right, we will finish human health and disease. That is what we are going to finish, right? Human health and disease. And after that we will start your class 11th syllabus, right? So as we promised, right, we are regular, we are consistent in this Game of NEET series, right? We didn't promise you something and we didn't, it's not like that we promise you something and we didn't complete it, right? So just look at the list of the chapter as well. But they all are the most scoring chapters. From human reproduction, yes, you can expect 2 to 4 questions. From reproductive health, 2 to 3 questions. Principles of inheritance and variation, above 4 questions for sure. It can be 6, it can be 7. From molecular basis of inheritance, again above 5. Okay, evolution 3 to 4, biotechnology. See, in 2022, in 2021, I guess 1 or 2, in 2021, right, 11 questions were there from these 2 chapters. Imagine, right? I don't remember the name of the year right now, maybe in 2021 or 2022. In 2021, 11 questions were there from these 2 chapters. 11 question means 44 marks, right? And we have completed this biotechnology principle and processes in 4 hours, right? There is a break time as well. So even if you will increase the speed now, you can finish that video in 4 hours, imagine, right? And for today's lecture, we need 3 hours or 3 hours, 15 minutes or 30 minutes. Okay, so our today's lecture is not going to be very, the duration is not going to be too much, right? Within 3 hours or within 4 hours, we will finish it. Okay, we will finish it. Okay, and if you want me to start the human physiology in class 11th, okay, fine, I don't have any problem, right? But I want you people to be consistent. So first of all, quickly hit the like button, everyone. Hit the like button, everyone. Yes, hit the like button, and if you're new to our channel, subscribe to our channel. And let me tell you something very interesting, people. So just have a look, right? Your NEET End Game 2.0 batch is here, and here in this particular batch, see, we are going to start it on 22nd of Jan. Right, on 22nd of Jan we are going to start it. And if you will ask me the zoology syllabus bache, so we'll be starting with the class 11th human physiology. So if you will join this batch now, you will cover the most important unit of your class 11th biology, and you can get this batch just at a price of 4499. And here in this batch, the proper mock test and the revision will be there. Now I know many of you must be thinking, ki ma'am, why this batch? You people are teaching us here, है ना? You people are teaching us here. You people are even having the special classes for the practice. Then what's special about this batch? Let me tell you. If you will join this batch, there will be the consistency in your preparation. Why? Because every day you know there will be the classes. Every day you know that a teacher is going to come. They will ask you the questions. They are going to teach you. So you will have a proper routine, right? You will know which thing you need to revise. Isn't it? Because we are at a point, we are at a stage bache, where we procrastinate. Okay, we get confused, we are overburdened with the syllabus, we don't know what to start, what to leave. Okay, we don't know the right amount of time that we need to devote to a particular subject or to a particular chapter. So if you will be the part of this batch, if there will be the proper classes of physics, chemistry, or obviously biology, then what is going to happen? Right, you will have a proper routine. Right, this is the first thing. Second thing is bache, quality content. If you will follow the classes of our batch, only that's what you need to revise. Okay, if you will complete that portion only, you are going to get very good marks in your final exam. Right. Third thing, the proper tricks will be provided to you. Okay, the proper tricks will be provided to you. No doubt, on YouTube also we teach you. We consider the NCERT, right, bache? But you know that here we have the limitation. This platform is for all, okay, right? So sometimes spammers are there, sometimes, you know, we have to limit many things, okay, because if I will start teaching beyond NCERT, you're not going to accept that. So there are some things which we need to explain. When we need to explain it, we need to go beyond NCERT. Okay, so everything you are going to get in this batch, everything you are going to get in this batch, proper batch and all, proper test and all, right, bache? So how can you join this batch? You can see the description box, right? You can see the description box, and here you can see this thing, NEET End Game 2.0. Click on the link, add your details, and you will get this batch. Okay, so be the part of this batch. Do not join it too late, otherwise there will be a burden of the classes, of the recorded classes. Okay, and you people are going to get the live classes here. Okay, live classes here. Done, bache? And secondly, you can see this, unlock the power of learning. Prices are dropped for your NEET UG subscription, even for your 2025, right? And plus you will get extra 5 months as well. So this is valid till January 18, and this is the coupon code that you have to use. Again, you will find the link in the description box. Okay, so let's start the biotechnology and its application. So tell me, what do you understand by the word biotechnology? Tell me, people, what do you understand by the word biotechnology? I started my last class with the same line. Okay, chalo, now quickly hit the like button and the subscribe button as well. Okay, hit the like button and the subscribe button as well. Class schedule. Acha, Dr. Strange, are you asking about this batch? But your classes will start from the morning till evening, so you have that evening time will be there for the self-study, bache, and sometimes it can happen that you will get the classes, you can say that physics, chemistry, biology classes are there. Sometimes might be for one subject you can get 2 classes as well. So scheduled is planned in this way. Okay, and guys, quickly hit the like button. We'll talk about the boards as well. So tell me, what about the biotechnology? Let's start, and please start with some, you know, fire emojis. It's too cold here. It's too cold here at my place. So that's what I'm expecting. Good. Thank you, Jivitha. Thank you so much. Thank you so much. Biopharmaceutical and the biological. Okay. Very good, very good, very good. So simply when it is the biotechnology, right, bache, so we know that basically we are going to mix the technique and the living organism. And why are we mixing the technique and the living organism? Simple, we need our benefits, isn't it? We do not have any other reason. The only reason is the benefits that we need. Okay, when it comes to the living organisms, not just about the humans, you can talk about the plants. You all have seen the plants. You know that various type of fruits we used to get from the plants, isn't it? Various type of fruits, the food, we are dependent on so many things. Same is the case of the microbes as well. Okay, same is the case for the microorganisms as well. There are many microorganisms that give benefit to us, right? In the last class also I told you, in when we discuss the old biotechnology, students, in the old biotechnology we only talk about the natural capabilities of the organisms, है ना? We only talk about their natural capabilities as in, right, natural capabilities as in, let's talk about the lactobacillus bacteria. So you know that this lactobacillus can make the curd. How it will act on that? What that lactobacillus will do? This lactobacillus is going to convert your lactose into lactic acid. It is going to convert your lactose into lactic acid, and this will act on the milk protein casein, and further coagulation will occur. So these are some natural, capabilities of the microorganisms. Okay, so in old biotechnology we cater that, but the point is, ma'am, why are you discussing it now? Why are you talking about these conventional ways? The simple thing is, I'm just giving you an example that look at these microorganisms, right? The way they are benefiting us. Okay, we are getting different acids, the antibiotics, right, other useful products, the bioactive molecules from these microorganisms. So when we mix technology with these living organisms, we want the benefits out of it, right? We want the benefits out of it. So here in the biotechnology principles and processes, we talked about the ways that how can we take that benefit, or how can we make the recombinant DNAs. Okay, how can we use the genetic engineering for all that things? But here in this chapter, because now you know the basic procedure, you know the basic process, here in this chapter it's only about the applications as in, you'll be talking about the golden rice, you'll be talking about the things like that. Okay, that is what we have to cover here. So how are we going to start? We are going to start with the 3 critical research areas of biotechnology, right? 3 critical research areas of biotechnology. So when you talk about that 3 critical research areas, bache, for the biotechnology, so it is to provide the best catalyst in the form of an organism or the enzyme. It can be anything, right? Basically right, when it comes to the 3 critical research areas of biotechnology, basically we want to improve that organisms, right? We want to improve that organism. We want some superior organisms. We need their product, we need their enzymes. Okay, so that's the first research areas, that is to provide the best catalyst in the form of improved organism, usually a microbe or the pure enzyme. Any doubt here? Any doubt here, Alina? Yes, Vijay, any doubt here? Second point is, okay, fine, you got the pure enzyme, or if you can say that you got that better enzyme, you got that better organism, whatever it is, right? Whatever it is. Now, what do we need? Okay, fine, you have the pure enzyme. Then what? Imagine if you are having a catalyst. You need a substrate, you need a reaction where you can use that catalyst, isn't it? Then only you can get the product. If you have a catalyst, you need the substrate as well, so that you can convert that catalyst into the, so that you can convert that substrate into the product. Okay, the substrate into the product. So here the point is, Vaishu, Krutika, right, Umesh, here the point is creating optimal conditions through the genetic engineering, or through the chemical engineering, for a catalyst to act. So the first thing is, we need the catalyst, we need the superior catalyst, we need the pure enzyme or the improved organism, then we need the conditions where that organism can act. Okay, where that organism can act. Are you getting it? Are you getting it? Right. And then finally, now see, you have the enzyme or you have the catalyst, you have the conditions. Ultimately, it will result in the production of what? The recombinant product. okay, or the superior product or the desired product, and you need to extract that. So that's why the third thing is downstream processing technology, so that we can purify that protein, we can purify that organic compound. So these are the three critical research areas. Okay, but today, right, when you talk about the biotechnology, there is nothing, there is no part which it didn't touch. Like you can talk about the therapeutics, you will see the medicines which are prepared by these recombinant DNA technology, or basically by using, you know, DNA, the DNA vaccines are there are different ways. Even if you talk about the agriculture, you will see genetically modified organism. You might know about the hybrid animals or genetically modified animals, or you can say that the genetically modified plants as well, right? So all the, like, in one way or another way, what is happening, we are trying to improve the organisms which are benefiting us, right? We want to get the best things out of it. Okay, so now when you talk about the agriculture, see, we have agrochemical based agriculture as well. Let's talk about the green revolution. You know that initially the yield was not too much, but after green revolution, right, after green revolution, what happened? The crop production, it increased, it enhanced, not just by 2 times, by 3 times as well. Okay, by 3 times as well, isn't it? Tell me, isn't it? When you talk about the green revolution, first thing is agrochemical based agriculture. It's very simple, and you might have seen that in the fields we use the pesticides, we use the insecticides, we use the chemical fertilizers. Why? Because we want the better crop, right? We want that crop to be healthy, isn't it? We want the yield to be more, isn't it? And even after green revolution, all that things happened, bache, right? That yield, it increased 2 to 3 times. Imagine what they did at that time, right? You know that they started using a lot of chemical fertilizers, the pesticides and the insecticides. Okay, it was valid, it was good, but still, if you compare our crop production with the population, okay, if you compare the crop production with the population, right, the demand is actually more, the population is more, so the demand is more. Are you getting it? So the, yes bache, so the demand is more. So now because demand is more, right, even because of green revolution, the crop production has enhanced, but still it is not able to fulfill that demand. Okay, the first thing. Now when you talk about, and yes, here, because chemical fertilizers, the chemical fertilizers are used, the insecticides are used, they are also resulting in the pollution, right? The soil pollution, the water pollution, right? The crop that is growing because of the use of excessive chemicals. You know that crop is also basically, what is it? Let's say if in the field I'm spraying that the fertilizer, that chemical fertilizer, right? If I'm spraying all that things, what is going to happen? The plant, they are also going to absorb that. That is valid? No, that is valid, right? It's a valid point. So the point is ultimately that crop is also not good, na, right? The food that we are getting, it is not also healthy, na, right? So now see the another way, like if we consider the production as well as the environment. So let's talk about the organic culture. So what is it? It is eco-friendly. Here we use the eco-friendly ways. Now when it comes to the eco-friendly ways, what do we have? Right, when it comes to the eco-friendly ways, then what do we have? It's simple, bache. Here in this organic culture, what's the story? What's the point here? Right, the biofertilizers will be used. Okay, bio-insecticides and the pesticides will be used. Okay, and why? Right, because we have to consider the environment as well. So no doubt it is a healthy way, but still it is not fulfilling that demand. And the third way is genetically engineered crop based agriculture, because this genetically engineered crop based agriculture, it is also, right, it is adding the superior plants or animals, right bache? And moreover, if I talk about the plants after harvest, the losses, they are less, right? We are getting we can make insect resistant, the pest resistant plants, right? So obviously our production will also not be affected. So that's the point. Okay, Laharika, that's the point, right? So now you can see this, the green revolution succeeded in tripling, right? The green revolution succeeded in tripling the food supply, but yet it was not enough to feed the growing human population. So demand is more, production is less, right? Demand is more, production is less. So increased yields have partly been due to the use of, right, improved crop varieties were used, and bache, better management practices were there. Agrochemicals like fertilizers and pesticides, they were used. Okay, so however, for farmers in the developing world, agrochemicals are very expensive. They are still expensive, right? So these things are very difficult to manage, right, bache? So it's not possible, right, that existing varieties, they can increase the production that much that it can fulfill the need. So yes, we have to look for other ways also. Okay, we have to look for other ways also. Now bache, this is what you need to keep in your mind. You know that from your you know that, right, in your NEET 2024 there are few chapters which are deleted. Yes everyone, Samiksha, Dr. Strange, Umesh, Veshu, there are few chapters. Yes, there are few chapters that are deleted. Hai na? Can you name the deleted chapters of class 12th? Can you just name the deleted chapters of class 12th? Quick. Quick. है ना, environmental issues, the strategies for enhancement in food production, right, reproduction in organism from class 12, they are deleted. So when we talk about the strategies for enhancement in food production, we used to discuss the topic of tissue culture, right? Because this chapter is deleted, now in new NCERT, they added the tissue culture part to the biotechnology applications, okay? So ultimately, the moral of the story is that we need to read the tissue culture in detail. Okay, we need to read the tissue culture in detail. Clear bache? And trust me, it's a very simple topic. Okay, what kind of topic it is? It's a very simple topic. And what's that? That is tissue culture, right? That is tissue culture. Okay, now let's talk about it. So first of all, you know that we all are developed from a single cell, and that single cell is the zygote. है ना, we all are developed from a we all are developed from a single cell, and that single cell is what? That single cell is the zygote. Why? Because of the property of cellular totipotency. Ultimately the word is totipotency. What's the word bache? The word is totipotency. So what is totipotency? Totipotency is the ability, right? Totipotency is the ability of a single cell. Right, it is the ability of a single cell to grow into a complete organism, right? To grow into a complete organism. That is what totipotency is. So now zygote is a totipotent cell, right? It is capable of developing into a complete organism. And when it comes to the tissue culture, na, this is the property which we are going to focus, right bache? When it comes to the tissue culture, this is the property that we are going to focus, okay, that we are going to discuss. Is that clear? Is that clear? Yes bache, is that clear? Okay, the tissue culturing. So Gottlieb Heberlent, right? I don't remember the spellings. Gottlieb, yes, that name you can check once. Heberlent, right? He was the first one who started this tissue culturing, right? He was the first one who started the tissue culturing. Okay, but he tried to culture the plant for the first time, Gottlieb Heberlent. Now the point here is, I told you about the totipotency, but how the totipotency is possible? What is the logic behind? See, it's very simple, bache. If you are talking about a human cell, you can pick up any human cell, you are going to get 46 chromosomes. Right? If you are picking up the diploid human cell, na, so definitely you are going to get that 46 chromosomes. Okay, all the chromosomes are present in that single cell, right? If you have that misconception that in the case of humans, right, our entire body, all the cells of our body, they have 46 chromosomes. It's not like that. Individual human cell is having 46 chromosomes if it is a diploid cell, right? If it is a diploid cell. Okay, okay. So now the point is that this diploid cell, it is having the chromosome, right? Chromosome is having that DNA, it is having that gene. So basically in all the cells genes are present, the complete information is present. It's just that bache, that with growth, with differentiation, with maturation, what happened? They become specified, right? What is the meaning of differentiation? Mr. Walkie Talkie, S, Veshu, Tomboy, what is the meaning of differentiation? When a particular cell, when a part, they will acquire a particular function. Let's relate it with anatomy of the flowering plants. Okay, let's relate it with the anatomy of the flowering plants. You remember anatomy? Yes, do you remember anatomy? So let's relate it with the anatomy of the flowering plant. Anatomy means internal structure. Okay, anatomy means internal structure. So you know that in the anatomy also, right, in the anatomy, what we discuss, we also talk about the differentiation or de-differentiation. Do not get confused if I'm using a new word. It is somewhere in biology. We talk about the differentiation exactly when it is the secondary growth. How do we start in the case of plants? That there will be the meristematic tissue. Slowly these cells will grow, right? They will differentiate. After differentiation, there will be the maturation. They will become specified. They will, right? They will become specified. They will acquire a permanent function. Right, they will acquire a permanent function. And bache, even when we talk about the secondary growth, right, there also we discuss now that firstly the de-differentiation occur, right? That is what we discuss now, that firstly de-differentiation it occurs, isn't it? Isn't it? That is what we discuss, yes or no? Yes or no? So basically all the cells, they have the ability, they have the information to grow into that complete organism. It's just that with maturation, right, the only particular genes they will express themselves. Okay, only that particular genes they will express themselves, not all the genes. Like if I'm talking about the neurons of my body or the muscle cells of our body, in that cells only that particular genes they are activated. Okay, only that particular genes they are activated, not all the genes they are activated, right? So that's the point. That's why this totipotency word is there. Now what about this totipotency in tissue culturing? Now scientists, they know about the totipotency. They know that if, let's say, if we take any part of the plant, okay, this is a plant, okay, this is a plant, you want to culture this particular plant. Okay, you want to culture this particular plant. So if you want to culture this particular plant, you can take any part of it. Okay, you can take any part of it for tissue culturing. Do you know that? Any part of it, right, for tissue culturing. And this is important, please pay attention. And the part of the plant, yes, and the part of the plant that is used for tissue culturing is known as explant. Do you know that? The part of the plant that is used for tissue culturing, it is known as explant. This is important, students. Please write down. This is important. That is known as explant. Okay. Okay, so what I just said, any part of the plant can be used for tissue culturing. So when it comes to the tissue culturing, you know about the ability, the totipotency will be used, right? We can take any part of the plant, we will call it as explant. Now what will happen in the lab, bache, in vitro, the sterilized conditions they are provided. Right, the sterilization should be there. Whenever you talk about the tissue culture, even if we are taking a part of the plant now and we are going to grow it, we should clean that part. We should clean that particular part with the chemicals so that it will become, yes, infection free. It should be without infection. So sterilization at every step is important. Okay, I'm not going to explain it again and again. Remember, sterilization at every step is important. Okay, sterilization at every step is very, very, very important when it comes to the tissue culturing. Okay, so this is the explant. So ultimately, let's say I'm going to take the shoot of this plant. So what should I do? Obviously, use your brain. Even if in the nature, if we are going to if we want a seed to grow, we have to provide it the conditions, the optimum conditions. Yes, yes. What are we going to do? Bache, walkie-talkie. Acha, now walkie-talkie is like, how can you consider the bark? Bark is a dead part. Bark is the dead part, right? Obviously, you need something which is having the genetic information, which is having that nucleus. Bark is going to be, we will consider it as dead. Okay, and right, it is going to be the dead. There are various conditions as well, right? Various conditions as well. We have to see each and every condition. We have to figure out that living cells. And moreover, bache, ultimately, Mr. Walkie-talkie, you have to go for the differentiation, right? I am just telling you that any part which is taken for tissue culturing is known as explant. Okay. Okay, I just said that any part which is taken for tissue culturing is known as explant. You can take anything. You are taking it, you are not taking it properly. I am saying that any part which is taken for tissue culturing, okay, you are going to call that plant, the part as explant. You are taking the leaf, you are taking the shoot, you are taking the root, you are taking the ovary, you are taking the anther, you will call it as explant. It is any part which will be used for the tissue culturing. Now, you have taken a shoot. Even in the nature, if you want to grow a plant, obviously you need to provide the proper care. The optimum pH should be there in the soil, the optimum temperature condition, the water should be proper, the essential medium should be there, right? The source of energy should be provided. These are the things, right? These are the things. So here also, if you have taken the part of the plant for the tissue culturing, right, which you are calling as explant, you need to grow that explant. You need to provide it the condition, the infection-free condition, and obviously you will be needing a culture medium for its growth, right? What do we need? The culture medium for the growth. Okay, okay, so ultimately we want that explant to acquire the ability to divide, right? From that explant we have to make the undifferentiated cell mass, right, Veshu? What we have to do? We have to make the undifferentiated cell mass. Clear, bache? So in this culture medium, exactly, you are right. Here in this culture medium where you are culturing your explant, what will it contain? It will contain all the essential nutrients, right? And in the lab you will maintain the optimum conditions. So here what should we add? The proper nutrients, the inorganic things, the organic things. Basically the plant growth regulators will also be there. Don't you think so? Right? Don't you think so? The plant growth regulators, PGRs, should be there. Can you name the PGRs that we need here? Yes, can you name the PGRs that we need here? Tell me. Can you just tell me the name? Yes, bache, can you just tell me the name of that PGRs that we need here? Yes. Auxin and cytokinins. Yes, exactly. So here in this culture medium or the nutrient medium, right, bache, you will be having inorganic salts. You can write down if you want to. You will be having the vitamins. Okay. Auxin, cytokinin, and obviously they should be in a proper ratio. Their proper ratio should be there. It's not like that you can use it in any amount. No, you need a proper ratio for it, right, bache? You need a proper ratio for it. And above all, there should be the source of carbon, right? Source of carbon for it, that is the sucrose, right? The carbon source, basically, for the fuel should be added here, right? The fuel should be added here. Just sucrose will be added. Okay, what will be added? Sucrose will be added. Okay, so because we are providing it a culture medium, we are providing it a nutrient medium. Now these cells, they will start their growth. They will start their growth. They will start their growth. And what will they form? They will form callus. What is the meaning of callus? Another important previous year question, bache. Right? Another important previous year question. Callus is the undifferentiated cell mass. Callus is what? It is the undifferentiated cell mass. Please pay attention on this word. It's undifferentiated. What is the meaning of, Kritika? What is, right, Desiti, what is the meaning of undifferentiated cell mass? That here cells are just dividing. They do not have the proper fate, right? They are not the mature cells. They are just dividing. Okay, so you are going to take the explant. It should also be infection free. You are going to grow it in a culture culture medium, right? Obviously optimum conditions are provided, optimum environmental conditions should be provided there, and slowly this explant will, because it is also utilizing that a nutrient medium, we have added the hormones as well, so the cell division will start, and with time what will we see? We are going to see the undifferentiated cell mass, undifferentiated cell mass. Clear? Right, bache? Now I will add one more word. Like when we also talk about the types of tissue culture. Do you know that we even talk about the types of tissue culture? So during that we also discuss callus culture, okay, and suspension culture. We discuss what? Callus culture and suspension culture. So please do not get confused here. Callus in general means undifferentiated cell mass. Now let's say for tissue culturing, when you use the agar gel, right, when you use the solid medium, right, when your culture medium is solid, ultimately your explant is going to make the undifferentiated cell mass, right? Right, it has to grow, okay, it has to grow. So basically when your medium is solid, right, when your medium is solid, you will call it as a callus culture, you will call it as a callus culture. And bache, when this nutrient medium, when it is a liquid, okay, it's not a it's something a gel like, it's liquid, then what will you call it? You will call it as a suspension culture. So do not get confused here, okay, do not get confused here. Clear bache? Clear bache? Is that clear? Is that clear? Right? So here medium gel with agar is provided. Okay bache? Okay bache? So it will have both type of regulators, right? In the callus culture you will see both type of regulators. You are going to see the auxin as well as the cytokinin. So basically you can say that such hormones like chemicals are used. ठीक है वैशु? Such hormones like chemicals are used. Clear bache? Clear, clear, clear. Right, right. So in the callus culture, this is another point that you need to focus. First of all, the culture medium is solid, right? Secondly, both the hormones, they will be used. Now in the suspension culture, usually what are they going to use? Mostly basically auxin is present here. Okay, so one thing is that your culture medium is going to be liquid, and the another important point is that it is going to contain the auxin. It is going to contain the auxin. So I will add one point here, right? Auxin 2,4-D. Anyone in the class who can tell me about auxin 2,4-D? I remember, right, Pankhuri ma'am, few days back she took the lecture on this plant growth regulator. What is the meaning of 2,4-D here? What is the meaning of 2,4-D here? Yes, anyone? Anyone? It's a VD side, Harvey side. What is D? What is D? This is my question. What is this D? Good Nandini. Good Nandini, very good Nandini. It's dichlorophenoxyacetic acid. Same way you have 2,4-D as T as well. Trichlorophenoxyacetic acid. Okay, okay. So remember this. So this is how in the callus culture the cell mass will form. In the suspension culture, right, that cell mass will form. But here in suspension culture, bache, proper agitation is there. Here in suspension culture, we do not want that cells to make the massive cell mass. We want to keep them in, keep them as single cells, and moreover whenever there will be the cell mass, it should be uniform. So in suspension culture the growth is more, right? Now I said that these are the hormones that we use. They can be used in different ratios, they can be used in singly. Can you tell me why, like how that auxin is going to help? Anyone in the class? How that auxin is going to help? What that auxin will grow? Root growth. Auxin is responsible for the root growth or for the shoot growth? Yes, auxin is responsible for what? Root growth or the shoot growth? Thank God, Akhankya. So yes, bache, when auxin is added, the roots will grow. Right, the roots will grow. But basically in tissue culture, what happened na bache? Let's say they have taken the explant. Firstly, the shoot growth is promoted. Okay, the shoot growth is promoted, and yes, you are right, cytokinin is used for that. Right, then it will be sent to the to a culture where auxin is there, and then what is going to happen? The roots will grow, right? The roots will form. So now when you have the root as well as the shoot, right? When you have root as well as the shoot, so basically root and shoot, they are the two important things for a plant. Root and shoot, they are the two proper things for a plant. So once you will get that root and shoot, after you have that plantlet, right, then you can propagate it, you can grow that plantlet outside as well, right? So this is how it works. Okay, this is how it works. This is something very simple. I'm not telling you that I have shared all the details of tissue culturing here. No, this is something basic that I'm explaining you here. Okay, this is something basic that I'm explaining you here, right? So shoot and the root formation will be there, and yes, you are right that auxin, जैसे कि your NNA and AA, it promotes the root formation. Cytokinins, they promote the shoot generation. Okay, right? And sometimes somatic embryos are also used for tissue culturing. Somatic embryo, we use the word embryoid for it. So anyone in the class who can tell me the meaning of this embryoid? Yes, what is the meaning of somatic embryo? Anyone? Yes, what root? Somatic embryo is the word. But it's very simple. See, these are the non-zygotic embryos. What are they? They are the non-zygotic embryos, right? Basically, you know that the embryo is going to form the zygote now. But here in vitro means in lab conditions, right? In vitro, what are we doing in vitro, bache? In vitro, from the somatic cells, okay, the embryos will be formed, right? So somatic cells, diploid they are. So from that somatic cells, the embryos will be formed. And how is it possible? We will use the chemicals. Just for an example, ammonium nitrate, right? Ammonium nitrate and auxin is used for the differentiation here, right? They are used for the differentiation here. Are you getting my point? So basically there are different ways by which we are getting different things, different parts. Now let's say if a plant is infected, okay, let's say you have a plant and that plant is affected, okay, it is having the virus infection, then what are you going to do? Tell me, what are you going to do? You want to grow a plant, but that plant is having what? That plant is having the viral infection. Then in that scenario, what will we do? Hanji, exactly. So meristematic, right? Meristematic tissue will be preferred in that case. Meristematic tissue is used for culturing virus free. Right, it is used for culturing virus free plant. Okay, it is used for culturing virus free plant. So yes, the meristematic tissue from the root tips, from the shoot tips, it will be taken. Now my question here is, right, my question here is that why that meristematic tissue, right, it is free from that infection, it is not having the virus? Why is it so? You are right. When there is the infection, the meristematic culture is preferred, right? The meristematic tissue is preferred. But my point is that why meristem? Why that meristem is not getting affected with the virus? And I want the answer in the comment section. Okay? I want you people to answer that question in the comment section. Is that clear? Is that clear? So the plantlets, they will be formed when they have few leaves. We can even transfer them to the soil, right, bache? Okay, so embryos can also be used for culturing, right? The different parts can be used for culturing. So as of now, this is what you need to know, right? This is what you need to know. Clear, bache? So bache, do you know anything about the anther culture? Right, then I will directly jump to the NCERT. Do you know anything about the anther culture? Yes, and the haploid production. Anther culture and haploid production. Anything about the anther culture? So first of all, tell me, what is that anther? What is that anther? Yep, Niharika, what is the anther? Yes? Anyone? Anyone? Male part? Okay, male part. That's it, that's all. This is what you know. But it's very simple na. Anther, yes, obviously anther culture, haploid, anther culture is used for the haploid production. So basically you are right, right? Anther filament, they are making the stigma, it is the part of microsporophyll, right? That anther and that filament, they are basically what? Stamen. So stamen is what? Right, you know that microsporophyll, right? Microsporophyll, the group of stamen, it is basically the andrecium. Right, it is basically the andrecium, that is what we know. So now here the point is, when you are doing the anther culture, right, you are growing that anther, obviously you will get the haploid plants, isn't it? You are going to get the haploid plants. So first time anthers, they were used to form the haploid plants of Dhatura, right? Dhatura. And you know why this? And sometimes because of this, by using the anthers, right, by using the anthers, we can also form the diploids. Can you tell me how? By using the anthers, we can also form the diploid. Because anther, when you will grow them in the culture medium, you are going to get the haploid plants, right? It is their ploidy. Okay, it is their ploidy. But I am telling you that sometimes from that anther, you can also get the diploid plants. How is it possible? The first thing is the wall of the anther, the outer coverings, the wall of the anther, right? That is also diploid. We can use that. And moreover, sometimes, bache, let's say this anther, it is haploid. It is haploid. By proper treatment, right, from this anther you can make homogeneous diploids, right? You can just repeat this, okay? You can just repeat this particular haploid set. Just if one set is there, anther, you can just multiply this, right? You can form its two sets. Just Colchicine treatment is given, okay Leharika? Colchicine treatment is given, okay? Okay. So haploids are used to produce homogeneous, or you can say that homozygous diploid, right? Homozygous means, see, this is a one set, you are going to double the another set. Colchicine treatment is given, related with cell cycle and cell division. Okay, Colchicine treatment is given. Right, Colchicine treatment is given. So you can relate it with the cell cycle. Clear, bache? So next is the somatic hybridization, which is given in the NCERT. Okay, next is what? Next is the somatic hybridization. So what do you know about the somatic hybridization? What do you know about the somatic hybridization? It is having another names as well. Yes, somatic hybridization. Of course, it will give you the hybrids, somatic hybrids. But what is this somatic hybridization? Anyone? But it's very simple. In somatic hybridization, you are taking two different cells, you are fusing them, and you are going to fuse their protoplast. So now here the question is, what is protoplast? Let's say you have taken this plant cell, right? You are removing its cell wall. You have a plant cell, you are removing its cell wall. So a cell, right? A cell without cell wall. A cell without cell wall is known as protoplast. A cell without cell wall is known as protoplast. So here in this somatic hybridization, you will take the protoplasts, right? These cells are of two different species. You are fusing the protoplast of two different species. Yes, bache, of two different species. So when you will fuse them, you will get the hybrid. Right, you are going to get the hybrid. Are you getting it? Are you getting it? So how are you going to fuse it? Obviously you should have fusogenic chemicals. I'll give you one very simple example. It's given in your NCERT, PEG. Right, I'm talking about the fusogenic substance. What are the fusogenic substances which will help in the fusion? Right, which will help in the protoplast fusion. So let's recall this process. What are we discussing? We are talking about the tissue culturing. Gottlieb Heberlein, right? He was the first one who started it. It is based on the totipotency of a cell. What is the totipotency? The natural ability of a cell to grow into a complete organism. It is a natural ability of a cell to grow into a complete organism. That is what totipotency is. Now here we can take the part of the plant for the tissue culturing. We can consider any study part, and we can call that any part which we are going to consider for the tissue culturing, it is known as explant, right? So obviously it should have the proper nucleus. This is the bare minimum that we need. Now we have to grow it in a culture medium, okay, the nutrient medium. Okay, the culture medium, the nutrient medium. So when you people talk about the culture medium or the nutrient medium, it is going to have inorganic salt. It will be having proper growth regulators, right? It will be having proper growth regulators. It will be having proper inorganic salts, and we will do all that things, right, in a sterilized conditions. The infection should not be there, right? The infection should not be there. We have to do all that things in a sterilized condition. That's the point, right, bache? That's the point. Now what is the next point here? The next point here is, right, you are going to take that explant, you will provide them the medium. Because of the hormones, the division will start, you will get the undifferentiated cell mass. So can you tell me in the chat section, people, and I really want each and everyone to answer here, please tell me that undifferentiated cell mass is known as, yes, please tell me that undifferentiated cell mass, it is known as that undifferentiated cell mass, it is known as callus. Exactly. What is it? It is callus. That undifferentiated cell mass, what is it? It is callus. Clear, bache? Clear, bache? Right? So it is what? It is the callus. Now what is the next point here? Now once that undifferentiated cell mass will form, obviously, let's say if you want to grow the shoot, the cytokinin will be provided, right? For the roots, auxin will be provided. Accordingly, right, the proper conditions will be provided. Once you will get a proper plantlet, let's say roots are there, shoot is there, few leaves are there, you can even shift it to the hard soil. It will further grow. Right, it will further grow. Now after that, there are different types of culture. Sometimes nutrient medium is solid, callus culture. Nutrient medium is liquid, suspension culture. We even use the haploids. We even can use the ovary for the culture, right, bache? As of now, we are talking about the somatic hybridization, where we are using the protoplast of two different species, right? We are using the protoplast of two different species. So protoplast of two different species, what is the meaning here, right? Protoplast of two different species, what is the meaning here? Protoplast is a cell which is without cell wall. Okay, protoplast is a cell which is without cell wall. So we are fusing them. And now if you are concerned that how can we remove the cell wall, it's simple. If you are using a plant cell, you know that for a plant cell you can use cellulase, है ना? That will dissolve the cellulose there. So enzymes are used to remove the cell walls, and then these two protoplasts, they will be fused. How can we fuse them? With the help of fusogenic substance. Right now we are talking about a chemical, that is PEG, polyethylene glycol. It is given in NCERT. Okay, so that's a chemical which is used for the fusion of these two cells, right? It is the chemical that is used for the fusion of these two cells. Is that clear? Is that clear? And even by treatment with, how can you fuse them? By treatment with calcium ions at high pH, that can also be done. And yes, we can also use the electrofusion, right? We can use the electricity, Nitesh, for the fusion. Okay, we can use the electricity, right, the electric shock for the fusion as well. So that is what you need to write down. Okay, we can use electric shock is also used for that fusion. That is also a point, right, bache? So once you will fuse these two protoplasts, what will you get? You will get a hybrid. You are going to call it as a somatic hybrid, right? So we have the example of that potato plus tomato, right? Bromato is also there. Brinjal plus tomato, bromato is also there. Okay, so these are the things that we have. Yes, definitely you will get the heterokaryon here. Okay, so any doubt? Yes, so we have to culture this fused protoplast of two different species. It is the heterokaryon, right? It is the heterokaryon, and you know that it will again regenerate cell wall. It will further grow. Fine, it will further grow. Okay, so obviously it is having the character of both the cells, right? It is going to have the characters of both the cells. Fine. Fine. So this is about the somatic hybridization, right? This is about the somatic hybridization, also known as parasexual hybridization. Okay, parasexual hybridization. And for the very first time it was tried for the case of tobacco, right? For two different species of tobacco plant, they were used, their protoplasts, they were fused, and obviously you will get the hybrid. Okay, you will get the hybrid. Okay, bache? So now tell me, if you are using a particular plant and you are making so many plantlets from that plant, don't you think that it will be just like a clone? Just in the case of somatic hybridization, you are using the protoplast of two different species. So whatever hybrid you are going to get, it will show the characters of both the species. But here, when normally you are taking the explant, you are growing it, obviously the new plants that are going to be, that will form, right? Right, the new plants that will form, they will be the clones. They are genetically totally same with their original parent plant. So soma clone is the word that we use. Very good, Samiksha. Soma clone is the word that we use. Okay, so now let's quickly revise it from NCERT. Here you can see, right, it's a suspension culture, right? Look at this, the steps here, right? Slowly that the cell mass will form. Okay, it will further grow by using the nutrient medium. Slowly see, or I know the pictures are not so clear, but try to, you know, visualize it a bit. See how that plantlets, how that plant is forming, right? The cell mass was there, how that cell mass is growing, because it is also getting the nutrient medium, all the essential things. Fine, bache. So look at this, it is actually a nutrient medium. This is a cell mass. You can see the cell mass, and from the cell mass, see how are they growing. Okay, so this tissue culturing can be used for the commercial production of the plants, right? The ornamental plants, when we want to make their multiple copies, okay, we can use one plantlet, and in less time we can form more plants, right? Right. So in different ways we can use it. Okay, sometimes let's say there is a plant which is on the verge of extinction, we can use the part of that plant, we can store all that genetic information, or we can, by a tissue culturing also, we can form more plants. Okay, so look at this part. We will discuss about the soma clones. So meristem or shoot tip, it was isolated. The shoot development is there, right? Because cytokinin is there. The axillary branching will come, right? Rooting of axillary formed shoots will be there. Sorry. Now then start from this part. Shoot tip is again isolated. Node isolated. Shoot development started. So cytokinin, it is promoting that shoot part. And further, finally, the rooting will be there. Okay, the rooting will be there. So it is the in vitro micropropagation. Okay, right? They have used the axillary bud method. This is just to show you. Okay, this is just to show you that how this works. So see, you took the aseptic explants, right? They should be infection free, right? This is the aseptic seedling. So surface sterilized. Right? They should be surface sterilized. Do you know the meaning of surface sterilized? Do you know the meaning of surface sterilization? It means, right, the explant, we need to clean that explant with the antimicrobial chemical. Okay, right. What is the meaning of surface sterilization? We have to clean that explant with the antimicrobial chemical. Okay, that should be infection free. Yes, prior to that, putting it on the culture medium. So we can also use H2O2, hydrogen peroxide, as the surface sterilizing agent, right? As a surface sterilizing agent, we can also use the H2O2. Fine. So see, aseptic explant is there. So we have to induce a dedifferentiation. What is the meaning of dedifferentiation? Related with anatomy here. What is the meaning of dedifferentiation? What are we doing here, bache? We are inducing dedifferentiation. Basically, we want that explant to start its division. So explant is the part that you have taken for the tissue culturing. Obviously, it is having the permanent tissue, but now you want it to divide, right, Umesh? Now you want it to divide. So if you want it to divide, you want the dedifferentiation, that we have to induce, right? See, you can see the cell culture here. So shoot regeneration is there, right, bache? Somatic embryogenesis, like these are different ways they are trying to show here. Regenerated shoots, right, bache? Regenerated shoots. This is a cell suspension culture. Okay, somatic embryos are formed here. Okay, somatic embryogenesis means somatic embryos are formed here. Okay, right? Done. So you just need to know about the, like, you just need the brief idea of this tissue culturing. So just let's read it from NCERT. So they are saying that as a traditional breeding technique, they failed to keep pace with demand and to provide sufficiently fast and efficient system for crop improvement. Another technology called tissue culture is also there, right? Just let's say there is a superior plant. Okay, you can directly take that superior plant, you can make many superior plants in the same time. Okay, in the same time by this tissue culturing. So see, what does tissue culture mean? It was learned by scientists during 1950s that whole plant could be regenerated from the explant. So what is explant? Any part of the plant taken out and grown in a test tube under sterile conditions in a special nutrient or the culture media. Okay, nutrient and the culture media. And the capacity to regenerate a whole plant from any cell, it is the totipotency. Clear, bache? It is the totipotency. Okay, so in nutrient medium there should be a carbon source such as sucrose. You know, sucrose, it's a disaccharide. Yes, sucrose, do you remember that sucrose? It's a disaccharide, it is the invert sugar, it is the non-reducing sugar. Yes, do you know in the phloem also, it is not the glucose, it is the sucrose that will be translocated. Okay, it is the sucrose that will be translocated. So see, inorganic salts, vitamins, amino acid, growth regulators, oxygen, cytokinin, they will be used. Fine, bache. So proper propagation of large number of plant will be done in a very short duration because of this tissue culturing. Okay, so because we are producing thousands of plants through tissue culturing, this is known as micropropagation. This is known as micropropagation. Any doubt? Hanji, any doubt? Please tell me, any doubt? Thank you, Afnan. Thank you so much. Thank you so much. Really happy to read that. So each of these plants will be genetically identical to the original plant, obviously, because you are making them from that particular plant part only. Now you're not changing the genes here, so that's why they are going to be same. You will also call them as soma clones. Okay, soma clones. So tomato, banana, apple, okay, they all are like, we are basically using them. Why? Because they are commercially important. Okay, commercially important. So another important application of this method is to recover healthy plant from the diseased plant, and that we can do by culturing the meristem. So this is important MCQ. It's a previous year question. Okay, Leharika, it's a previous year question. Fine. So this can also be asked in your final paper. You can write down that scientists have succeeded in culturing meristem of banana, sugarcane, potato, etc. So if I will be the examiner, I will definitely try to confuse you. I'm going to put such question that you people ignore. Okay, that you people ignore. Okay, so meristem culture is required. Meristem culture is used to form the plants which are free of virus, right, to form the virus free plants, and scientists, they have succeeded in culturing meristem of banana, sugarcane, potato, etc. Okay, banana, sugarcane, potato, etc. Done, bache? Done. So scientists, they have even isolated, right, they have even isolated single cells from the plants, and after digesting their cell wall, they have been able to isolate naked protoplasm. So we have already discussed the somatic hybrids. I hope the somatic hybrid and somatic hybridization is clear to you. Okay, so tomato, so obviously you will get a plant which is having, if it is a tomato, it is having the characters of potato and tomato both. So you cannot say that, because some genes, they will also be missing, so we cannot use it commercially, but yeah, still this can be done. Okay, right, because they do not have the desired combination of the characters for commercial utilization. Okay, but still we can form the somatic hybrids like this. So any doubt, bache? Any doubt here? Yes. So now let's talk about the genetically modified organism. So if in simple words I have to explain it now, so I'll simply tell you that basically they just took the tissue culture topic because they don't want to delete it. It is one of the most important topics. That's why they added it in biotechnology. Okay. Okay, that's why they added it in the biotechnology. ठीक है? So how are you feeling? Are you sleeping properly? Hanji, are you sleeping properly? Because these days this is what I'm feeling, that biology class, this is not for revision or something, right? You are using this class to sleep because we only want to read the physics or the chemistry because biology is easy, है ना, biology is easy, ma'am. We will get 360 out of 360 in biology. There is no need to read the chapters like biotech, like molecular basis of inheritance and molecular basis of inheritance. I was stupid who was taking two classes for that particular chapter. I don't want to compromise the content, right? My students should understand everything, right? There are many students, those who are only dependent on the YouTube classes, right? So I should think about them, this, that. And when it comes to student, they don't even bother to write a comment. They don't even bother to write a comment. They don't even bother to revise that particular chapter. Ma'am, is it virus resistant plant or virus free? It's Veshu virus free. The plant, one plant is infected, right? You want to make the healthy plant from that infected plant. That's why you use the meristem, because in meristem that viral infection is not there. The virus is not there in that meristem. Ma'am, how to feel more confident in the NEET exam? Bache, first of all, obviously the syllabus completion is going to help you when you really want to feel confident, but it is not only the syllabus completion. Sometimes it is our general human tendency that we are always skeptical about the things, okay, or we are not we like people like me, even if I'm prepared now, I don't feel confident. I have that anxiety problem or something, and definitely I'm going to spoil it, ruin it, like this. So for the NEET, all I can say is that please complete the syllabus, please practice the questions, please analyze the mock test. Okay, when you will analyze the mock test now, you will get to know about your mistakes. You should make the error notebook, you should write down that errors, okay, and especially the errors that you are making again and again. These things are important. Okay, these things are important. Okay, ma'am, you don't reply us. We comment. I read all the things. Right, I read all the things, Learika. Chalo. So now let's talk about the genetically modified organism. But I'll say one thing, only one thing. Okay, and that thing is keep, please do not waste your time. It's very difficult to start again. It's very difficult to be consistent again. Okay, if it's your first drop year, if you're thinking to go for a second drop, you can. I'm not saying it's not, but then that journey, it becomes very hard, right? Because there will be many sleepless nights now. There will be many sleepless nights now when you will just think about the NEET syllabus, NEET exam, right? Okay, and you'll try to, you'll just and I have seen that now, I have experienced that after the exam, what we feel, I wish I could go back, है ना? I think this is what we all feel. I wish I could go back. Arey yaar, I marked that answer incorrectly. I know the right answer. At that time I made the mistake. I wish I could rectify it, है ना? So things like that, they happen. So basically, please follow a proper strategy, be consistent, and practice as much as you can. Practice as much as you can. Okay, right now my special class link is not there in the description box. Okay, but soon you will get it in the description box. Okay, you people will get that after the class, you will get that link in the description box. So all I can say is that please join your class tomorrow, in which we will practice the questions from the entire biotechnology unit. This is the first point. Okay, this is the first point. And second thing is, bache, this batch, this is the ultimate batch for the revision, that is your NEET Endgame. We are starting, we have launched NEET Endgame 2.0. We are going to start the first class from 22nd of January, and you will get the live classes. Okay, just let's say in NEET Endgame we have finished 1 or 2 chapters. That chapters, you will also learn that chapters, but at the end, once we will finish the syllabus, it will be like this. it will be like this. Okay, okay, so be the part of this batch before it's too late. Now come back to the topic, the genetically modified organisms. But we also use the word transgenic plants, isn't it? We also use the word transgenic plants, transgenic animals, plants, or transgenic organisms. What is the meaning of transgenic? What is the meaning of transgenic? Ultimately it is genetically modified. Ultimately it is genetically modified, है ना? So in transgenic organisms, basically they are the one having the foreign gene. Okay, they are the one having the foreign gene. What are the transgenic animals? They are the one having the foreign gene. Now when here see, it is given in NCERT, genetically modified organism, and for this particular chapter there is no need to go beyond NCERT. That's why I'm taking the NCERT screenshots here, right? So here these GMO, they made crops more tolerant to abiotic stress, obviously by gene modification. Why we are in agriculture, why we are more concerned about these genetically modified organisms? Because here we can add a desired trait, we can remove the undesirable trait, we can make that plant resistant to abiotic stress, like it should have, we can make it more tolerant to the temperature changes, right, to the pH changes, like this. This is the meaning of abiotic stress here, right? Cold, drought, salt, heat, like this. Okay, so it reduced reliance on chemical pesticide. What is the meaning of this line? Pest resistant crop. I hope you remember the example of BT cotton. We will discuss about it in detail, right? I hope you remember the example of BT cotton. BT, Bacillus thuringiensis. Okay, it's the example of pest resistant crop. So basically when we have introduced a gene in particular plant that is making it pest resistant, we know that there is a crop, that crop is having its particular pest, है ना? There is a crop, that crop is having its particular pest. Okay, so by introducing the genes there, okay, by introducing that proper pest resistant gene there, what can we do? What can we do? We can, right, what can we do? Pest resistant crop we are talking now. So what can we do, bache? We then, because it is already pest resistant, there is no need to put extra fertilizers there, right, extra chemical fertilizer there. So it will also reduce the post harvest losses as well. And moreover, bache, we can make our plants to utilize, right, the nutrients from the soil at a very with a very better efficiency. We can make such plants as well. Okay, and then here you can see enhanced nutritional value of food, and bache, that is known as biofortification. Please write down this word, biofortification, right? When there is the improvement in the nutritional value of food, the process of improving the nutritional value of food, it is known as biofortification. So there are various examples. The best is the golden rice, which is a source of vitamin A, right? So it can be given to the people, it can be given to the humans having low vitamin A, right? So the foods having high nutritional value, right, this is one example, is golden rice. There are many other examples also in which they are some particular amino acid enriched, some particular protein enriched. Okay, so the point here is, right, so that process of enhancing the nutritional value of that food, it is known as biofortification. What is it? It is known as biofortification. Clear, bache? Is it clear? Yes, Okay, so in addition to these, GM has been used to create tailor-made plants to supply alternative resources to the industry in the form of starch fuels and the pharmaceutical. Right, bache? I'll give you the example where from the plants we can take the seeds which are having particular chemicals, right? Basically in different plants we can introduce, even in the animals we can introduce few genes that will make their products more nutritious. Yeah, they will have proper chemical that can be extracted, and the large scale production can be done by this. Okay, so now let's talk about the Bt toxin. Okay, let's talk about the Bt toxin, the important topic. So basically in this particular chapter, that Bt plants Bt toxin part, Melodigyne incognitia part, gene therapy, genetically engineered insulin, 4 main topics, right? And now it is the tissue culturing as well. Okay, now it is the tissue culturing as well. So basically there are 5 important topics, and that topics like biopiracy, right? Biopiracy, biopatent and all, they are very simple topic. I don't think that you need to use a lot of brain for that topic. Okay, but these topics are very important. Your Bt plants, right? Your genetically engineered insulin, your gene therapy, and this RNA interference, basically Melodigyne incognitia, and this now is your tissue culturing. So do not leave the topic of tissue culturing. Fine. So before starting the Bt cotton, let me quickly remind you about the Agrobacterium tumefaciens. Why am I telling you about the Agrobacterium tumefaciens? Because here in this particular chapter we are going to talk about the examples, right, where we will be using what? Where we are going to use this particular, yes bache, we are going to use this a particular plasmid. Okay, so Agrobacterium tumefaciens. Okay, Agrobacterium tumefaciens. So basically it's a soil borne bacteria. What type of bacteria it is? It's a soil borne bacteria. Okay, what type of bacteria it is? I'm repeating it again, bache. It's a soil borne bacteria. This bacteria, it used to infect the dicot plants, right? It infects the dicot plants. So what this Agrobacterium tumefaciens is going to do? See here, because this is a bacteria, it is having this nucleoid and it is having this plasmid. We call this plasmid as Ti plasmid. This plasmid is known as, yes bache desti, this plasmid is known as Ti plasmid. Why do we call it as Ti plasmid? Why do we call it as Ti plasmid? Ti is tumor inducing. Ti is tumor inducing, right bache? Here in this particular plasmid, if you look at this diagram carefully, okay, if you look at this diagram carefully, so here in this diagram, okay, here in this diagram, see this, Ti plasmid it is, right? Ti plasmid it is. So it is having the tDNA. Can you see this? It is having the tDNA. But this particular bacteria actually here in this bacteria this tDNA is the one which can make normal cell cancerous, right? This tDNA is the one which can make normal cell cancerous. Right bache? Right bache? So as I said, it is going to infect the dicot plants. Dicot plants having, broad leaves. Dicot plants having broad leaves. For an example, soya bean. Right, there are many examples, but as of now I'm giving you only one example, that is soya bean. So basically this bacteria, its tDNA, it is the one which can convert the normal cell into the cancerous one, and due to which that dicot wait, let me try to draw that diagram, but obviously it is not. Okay, so here this plant will get what? It will get crown gall tumor because of the infection of this plant, the disease crown gall tumor will occur. Okay, crown gall tumor, it will occur. Okay, so it is ultimately the tDNA of the plasmid that will convert, I'm repeating this again, that will convert normal cell into cancerous one. Fine, it will convert normal cell into the cancerous one. Understood? Understood, right? So scientists were like, let's use the natural capability of this organism. It's a natural engineer. You know, this bacteria is known as natural engineer. Why? Because it knows how to convert one cell into another, how to convert normal cell into cancerous one by sending its tDNA. So scientists, they were like, theek hai yaar, it's good. This bacteria is so capable, right? It can convert that cell. So they were like, let's change this tDNA. Hai na? Because when they studied this plasmid, they got to know that here in the tDNA there are some recognition sites, right? Here in this tDNA there are some recognition sites. What is present there, Niharika? Some recognition sites are present there in this tDNA. Okay, some recognition sites are present there in that tDNA. So by using the particular restriction endonuclease, we can remove this tDNA, right, bache? We can insert our own gene of interest. Okay, we can insert our own gene of interest. Remember insertional inactivation? Anyone in the class? Do you remember insertional inactivation? Like here in tDNA, when you know about the recognition sequence, right, you will use a particular restriction endonuclease, you will make the cuts there, right? So by creating the space there, you can put your own tDNA. You can put your own desired gene, desired DNA, and what is going to happen? We will get this recombinant plasmid. Okay, we'll get this recombinant plasmid. Okay, and that recombinant plasmid, it is basically harmless, right? Now this bacteria is working as our agent. Okay, that bacteria was naturally capable of transferring its tDNA into that plant, no, but now it will transfer our gene of interest. Okay, that is what we are going to use here. This is the vector that we are going to discuss here. So this is the example of genetic transformation in plant. Fine, this is the example of genetic transformation in plant. So that tDNA, instead of that new DNA, will be added with a desired trait, with a desired new gene, and further that gene will be passed to that dicot plant. So that's how it works. Okay, that's how it works. So the next thing is the BT plants. Okay, next thing is what? BT plants. So now what is the meaning of this BT? Tell me. What is the meaning of this BT? Bache, B stands for, this B is from Bacillus, T is from Thuringiensis, right? B is from Bacillus, T is from Thuringiensis. So Bacillus thuringiensis, that is the BT, right? That is the BT plants, Bacillus thuringiensis. So basically it is Bacillus, means a rod shaped bacteria. You know na, Bacillus word is associated with the shapes of bacteria. So can I say that it's a rod shaped bacteria? Actually, bache, this particular bacteria during its lifespan, it produces a toxin, right? It produces a toxin. That toxin is an insecticide, right? That toxin is an insecticide. What's the point? That this particular bacteria during its lifespan, okay, this particular bacteria during its lifespan, this bacteria produces a toxin, and that particular toxin is an insecticide. But now here, ma'am, the question is that if it produces a toxin, why that toxin is not destroying this bacteria? Why that toxin is not destroying that bacteria? Because that toxin is a prototoxin. Now what is the meaning of prototoxin? It is the inactivate form of a toxin. This toxin is not activated, it is inactivated, right bache? It will only get activated in its particular host, right? The cell, the organism that it is going to destroy. Clear bache? Clear bache? So we are going to talk about the BT plants, which are the example of these are the insecticide plants. Okay, or you can say that not exactly the insecticide, my bad, pest resistant plants, insect pest resistant plants they are. Fine, in pest resistant plants they are. Fine. So prototoxin, it is the inactive form of toxin. Now this bacteria, it used to do that. This bacteria, it produces the toxin. And why? Because of the genes. So basically scientists, when they studied the this bacteria, So they figured out that there are various genes that are killing particular insect pests, and it is not only about the insect pests. It is not only about the insect pests. These genes, they are also, there are some nematodes, there are some genes that can also kill the nematodes. Okay, but in your syllabus there are only that cry gene, na? That is what we discuss. But otherwise, this bacteria, from these various bacterial strains, we can also get the genes that can also kill the nematodes, that can also kill the beetles. Okay, but this is not nematode root, Umesh, that is different. Fine. So this is the speciality of this Bacillus thuringiensis, that it produces the toxin, that is the insecticidal. And now even in, you know, even in the fields, right, for your crops, in the market, the sachets in which the spores of Bacillus thuringiensis, they are present. Right, in the market there are sachets now where, you know, spores of this Bacillus thuringiensis, they are present. So that spores, they will be mixed with water, right, and then you know, that will be sprayed in the fields. Okay, and that will also destroy that particular pests, their caterpillar. It works like that. Now how are we using it for genetic engineering? Okay, the point is, how are we using it for genetic engineering? Now the point is, let's say if I will take out that particular gene from the bacillus, the gene name is cry gene. The gene name is cry gene. So when you write the name of the gene, okay, when you write the name of the gene, it should be in small letters, in italics, or it should be in small letter. So if it is a cry gene, it will form a cry protein, right? And for protein, the first letter will be in capitals. Okay, the first letter will be in capitals. So we have the cry gene and then we have the cry protein. So for cry protein, the first letter will be in capitals. That is the point that we have. Now what will we do? We will take out that gene from this bacteria. Let's say this is the bacteria. Okay, we isolated the gene from that bacteria. Now we will introduce that gene. We are going to introduce. This is what we are going to do in this genetic engineering, right? In this genetic engineering of this pest resistant plant. So we will introduce that gene in this particular plant. Now can I call this plant as transgenic plant? Can I call this plant as transgenic plant? Yes, tell me. Can I call this plant as transgenic plant? Of course I can, because it is having the foreign gene. Now bache, whenever any let's say any pest, let's say it is the caterpillar of that pest. Okay, it is the caterpillar of that pest, that insect pest. Don't judge my diagram. Don't judge my diagram. So that pest is, right, that caterpillar of that, right, this caterpillar is like, today I'll have party, right? I'm going to destroy a particular plant, I'm going to destroy a particular crop, I will have a proper party. So now this caterpillar will go, right, and that caterpillar is going to eat this particular plant, its leaves. Now what is going to happen? You know, now it is having a cry gene. It is having a cry gene. Cry gene is going to form the cry protein. No doubt initially it will be inactive, but once this caterpillar, it will ingest this leaf now. So here in the body of this pest, alkaline pH is there. That's the point, bache. Alkaline pH is there. Basic pH is there. Okay, Desti. It is what type of pH it is? It is the basic pH. Alkaline pH is there. And because of this alkaline pH, that prototoxin, that inactive toxin, it will get activated and it will solubilize here. Right? Because of the alkaline pH, that prototoxin, right, it gets activated and solubilize here. Solubilize in the gut. So now after the activation, what is going to happen here, bache, after the activation? Because now the toxin is activated, right? That toxin is activated. So now in the, let's say, you know, na, gut, alimentary canal, gut. So now in the midgut epithelia, right, in the midgut epithelial cells of this insect pest, now this toxin is going to create the perforation. Just imagine this is the gut. Due to alkaline pH, because our toxin, it is activated. Okay, so now this toxin is going to create the pores here. it is going to create the pores here in the midgut epithelium. The pores will be created. So now because imagine like, let's say in your guts, sorry, in your gut, if there will be the pores, imagine what is going to happen, right? Obviously the water from the surrounding will enter in the cell, है ना? Because pores are there, so water from the surrounding will enter here, so there will be the swelling. So there will be the swelling of the cell, and ultimately it will result in the lysis also. It will result in the destruction of that cell also. And this particular insect pest, it will die due to starvation, right? It will die due to starvation. Are you getting it? It is going to die due to starvation. Is that clear? Is that clear? Yes, is that clear? Right? So that is how this dangerous, this harmful, this cruel toxin work. So Bacillus thuringiensis, which is just a microscopic cell, which is a bacterial cell. Imagine how dangerous that bacterial cell is. है ना? How dangerous that bacterial cell is. That bacterial cell is like, ठीक है, don't worry, I have the solution. I will give you the gene, you insert them in the plants, you make them pest resistant, because any plant pest, when obviously the specific plant pest, when that specific plant pest will feed on that transgenic plant, the toxin is going to kill that pest. Okay? So the pores will be created in the midgut epithelium. Understood? Sure? Done? So do not forget the names of that genes. Okay, do not forget the names of that gene. Can you tell me again? I'm repeating, bache. Cry gene, it will give you cry protein. That's how you have to write it. The nomenclature is important. So we have cry1AC and cry2AB. So cry1AC, cry2AB, it will control, yes bache, it will control cotton bollworms, है ना? Cotton bollworms, right? So because now cotton bollworm, it will not be able to destroy the cotton balls, so we will get the mature full cotton balls, right bache? Right? So one is cry1AC and cry2AB, and the second one is your cry1AB, right? Cry1AB, and that cry1AB, it is going to kill corn borer pests, right? So it is for the cotton, it is for the corns. It is for the cotton, it is for the corn. So this is important. This information is important. So that's how your BT plants, they'll be created. Fine. That's how your BT plants, they will be created. So BT is the short form for Bacillus thuringiensis. Okay. So BT toxin is produced by bacterium. It is having, the gene has been cloned from the bacteria. It has been expressed in plants. It will provide resistant to insect. Okay, so obviously it is the biopesticide, right? It is the biopesticide. What is it? It is the biopesticide. Okay. Done? So now this information is important. Some strains of Bacillus thuringiensis, they will produce proteins that kill certain insects like lepidopterans. See bache, this is what I always highlight. You know that when you read about the animal kingdom, in the animal kingdom, right, phylum Arthropoda, you know, it is the largest phylum, and here in this particular phylum we have class. Which class is there? Class Insecta is there. Which class is there? Class Insecta is there. So in class Insecta there are different orders, right? In the class Insecta, you know that there are different orders, just Lepidopterans are there, Dipterans are there, Coleopterans are there, right? So now you must be thinking, ma'am, why are you linking this topic to the animal kingdom? Sometimes now we get questions like this, or we are not able to recognize. Okay, let's say if they will ask you, give me the example of the order Coleopterans. Out of the following, which examples, which organisms they belong to the order Coleopteran? Okay, and we are not able to recognize such things. And what we do, ki, arey yaar, this part is from the animal kingdom. It is something extra. It is beyond NCERT. It's not like that. Okay, so whenever you are preparing for any of the competitive exam, just keep one thing in your mind, right? That you are not studying class 11th biology. You are not studying for class 12th. You are studying the entire biology. Okay, it should not be 11th biology, 12th biology, it should be entire biology. It should not be botany, zoology, it is biology. Okay, it is biology. Fine. So Lepidopterans, like your tobacco, budworm, armyworm, Coleopterans, the beetles, and Dipterans, flies, mosquitoes. Okay, Dipterans are flies and mosquitoes, right? So as I said, they will produce the protein crystal during a particular phase of their growth. This crystal contains a toxic insecticidal protein, right? So this toxin protein, it exists as an inactive prototoxin, right? So once an insect ingests the inactive toxin, it is converted into an active form due to alkaline pH. So it is the alkaline pH of the gut that will activate that toxin, which solubilize the crystal, and activated toxin will attaches to the surface of midgut epithelial cells, and it will create pores that cause cell swelling, lysis, and eventually the death of the insect. Fine. So here you can see cry. So cry 1 AC, cry 2 AB control cotton ball worms, and cry 1 AB corn borer. Important it is. It's very important, right? It's very important. So see, cotton ball, they are B. In the case of B, they are fully mature cotton ball. Okay, they are fully mature cotton ball. Done. So next is the pest resistant plant. Okay, next is another pest resistant plant that has formed by this RNA interference. So now what we need to discuss? We need to discuss the RNA interference, right? We need to discuss the RNA interference. But the session we started at 7:10 or 7:15 now, so it will go till 10:30 or 10:15, 10:30. Bas. See, RNA interference, main topic. Genetically engineered insulin, main topic. Gene therapy, main topic. Then molecular diagnosis is there, right? It is not in that detail here, so obviously, right, there is nothing to worry about there also. And then what? Bas, then biopatent, that biopiracy, that's it. That's it. So I really don't think that we need a break, है ना? I really don't think that we need a break. What do you think? What do you think? Do you need a break? Tell me, do you need a break? No, na? Acha, dinner is waiting for you people. Chalo, then I'll give you a break for 15 minutes. Okay, then I'll give you a break for 15 minutes. So today I'm not well. I really don't want to stretch this topic. Okay, but it's just that I don't want to cancel the marathon, and the session is not so lengthy. So that's why I thought that I should take it. Even I'm not feeling well. So it's a request. Please be consistent, right? So we have, right now it's 8:50. So 8:50 to 9:10. That's your break time. Please be charged, be energetic, and then we'll continue this. And even if we will start it at 9:10, we can finish it till 10:30. So there is nothing to worry about. Okay, there is nothing to worry about. So please join back soon, and then let's continue. Okay, so RNA interference, genetically engineered insulin, gene therapy, molecular diagnosis, bas, 4 very important topic. That biopiracy and all, they are very, very, very easy. Okay, chalo. So before this dinner break, just tell me about GEAC. What is GEAC? Let me see the revision as well. What is GEAC? What is GEAC? What is GEAC? Anyone? No? Chalo, we'll tell you after the break. Okay, so let's start the class, everyone. So done with dinner? Okay, let's start the class. Fine. So now what are we going to talk about? We are going to talk about the transgenic tobacco plant, right? That is what we need to discuss. Okay, and for understanding the transgenic tobacco plant formation, we should know about the RNAi. This is what we are going to discuss. Okay, so quickly share the session link with your friends and invite them that ma'am has restarted the class, so everyone be the part of this class. So what we need to understand now, we need to understand the RNA interference. What we need to discuss, or we need to understand the RNA interference, that is RNAi. The very first point that you need to know here is that it is a cellular defense mechanism. It's a cellular defense mechanism in eukaryotes. Okay, be very specific, bache, right? It's a cell protective mechanism, cellular defensive mechanism, or the protective mechanism in the case of eukaryotes. You are not going to see it in the case of prokaryotic cell. Are you getting it? You are not going to see it in the case of prokaryotic cell. It's a protective cell mechanism or cellular defense mechanism in the case of eukaryotic cell, that is RNA interference. Now see, the word is RNA interference. Ultimately, even when you are talking about a eukaryotic cell, what do we have? We know that in the nucleus there will be the transcription, and in the cytoplasm there will be the translation, isn't it? In the nucleus there will be the transcription, and when you are talking about the cytoplasm there will be the translation. So ultimately, whenever we talk about the expression of a gene, right bache, whenever we talk about the expression of a gene, bache Samiksha, what is going to happen? A gene is going to express itself by making the protein. Now I'll give you one example. Let's say let's take the example of a nematode, and that is what we have to discuss even in the case of transgenic tobacco plant. That is what we need to discuss as well. So nematode, what are nematodes? First of all, tell me, what are nematodes? Do you remember nematodes? Right, they belong to phylum Esculminth. Nematodes. Okay, so we have one nematode that is known as Melodygine incognitia. The name of the nematode is Melodygine incognitia. That is the name of nematode. But this nematode, it decreases the production of the yield of tobacco plant, right, the yield of tobacco plant by infecting its root. That's how it works. Okay, by infecting its root, right? Do not get confused ki ma'am, you were talking about this RNA interference, then why are you talking about the nematode? I'll tell you. See, gene is going to express itself by making mRNA, one concept. Secondly, right down, we need to talk about the transgenic tobacco plant, and that transgenic tobacco plant is someone, that transgenic tobacco plant is the one which is resistant to this nematode, right, which is not letting this nematode to affect itself. Now how these two things are related? They are related because of RNA interference. The word is RNA interference. So here bache, RNA interference means we are not letting a particular RNA, our target RNA, right, we are not letting a particular RNA, our target RNA, right, our target RNA to form a protein, right? How are we going to stop the expression of a gene, right? How can we stop the expression of a gene if we will not let that gene form the protein? If we will not let that gene form the protein, if we stop the formation of this particular protein, this gene will not be able to express itself. Is it valid? Is it right? Right, is it right? So this is something we are going to do in RNA interference, right? In RNA interference, we will have a target, right? We will have a target mRNA, and we will not let that mRNA to form the protein, right? We will not let that mRNA to form the protein. That is what we have to do in the RNA interference. Okay? So for understanding this RNA interference, or for this RNA interference, we need double-stranded RNA. We need the enzyme which is known as dicer. This enzyme is actually an endonuclease. This dicer is nothing but it is actually an endonuclease, right bache, right? It is RNAs, it will digest RNA, and RISC, RNA induced silencing complex, or basically siRNA, small interfering RNA. That is what we need for the RNA interference. Okay, that is what we need for the RNA interference. Clear bache? Clear bache? Then how are we going to achieve this feat? Let me tell you. See, very simple it is. Array. Very simple it is, right? It is very simple. Just okay, it is very right, it is very simple. See, now there is a nematode, right? It is pathogenic. There is a nematode, and this nematode is what? It is pathogenic. It is pathogenic on tobacco plant. You tell me one thing, how can it infect the tobacco plant? How can it infect the tobacco plant? Like, obviously it must be having a gene that will make a protein, है ना? How can it infect a tobacco plant? How can it infect a tobacco plant? Obviously because of some proteins, because of some protein. Please try to understand this. Try to understand this, Kevin. I have a nematode, it is pathogenic on tobacco plant, right? I have a nematode, it is pathogenic on tobacco plant. Okay, this diagram is no doubt very bad, but ठीक है, we can use it to understand our topic. I have a nematode which is pathogenic on tobacco plant. Let's say here I have the roots of that tobacco plant. How this nematode is going to infect this tobacco plant? How that nematode is going to infect this tobacco plant? How this nematode is going to infect this tobacco plant? This nematode na bache, this nematode is basically, it is having some genes, right? That gene within this cells na, right here, that gene forms a protein, and that protein is actually decreasing the yield here, decreasing the production here. Try to understand this point. Try to understand this point, right? Whatever you have read about this topic, ki nematode specific gene, agrobacterium, forget about everything. Try to understand the procedure here. I am talking about RNA interference. I am telling you that it is a cellular defense mechanism in the case of eukaryotic cell. It is a cellular defense mechanism in the case of eukaryotic cell. Now here in this particular RNA interference, we will be having a particular target mRNA, and we will not let that mRNA to form the protein. How are we going to achieve it? This is what we need to focus, right? This is what we need to understand. Okay, this is what we need to understand. So I'm giving you one example. I'm telling you that here I have a nematode. This nematode infects the tobacco roots. How? Because this nematode is having certain genes that make the protein, and that protein is infecting this roots of tobacco plant. Now let's say if this gene, if it will not form protein, do you think then it can destroy or it can infect the roots of the tobacco plant? Tell me. If this gene, if it doesn't form protein, if this gene, it doesn't form protein, right? If this gene, of this particular gene, if it doesn't form protein, so do you think that it can infect the roots of the tobacco plant then? Of course not. Of course not. Definitely not. So if I want to make a transgenic tobacco plant, so here in that transgenic tobacco plant, I have to make sure that this gene, it doesn't get expressed. Right, that this gene, it doesn't get expressed. This part clear? This part clear? Now how to do that? Just say, let's say for this, whatever is your target mRNA, right? Whatever is your target mRNA. Now understand in general what is this RNA interference. So whatever is your target mRNA, just say, let's say this is my target mRNA, right? Obviously I need the sequence of this target mRNA. Okay, I need about this target. I know about this target mRNA. So bache, I am going to introduce, right? See, it is a cellular defense mechanism in eukaryotes. Do not consider the prokaryotes here. So I am going to introduce in that particular eukaryotic organism, I am going to introduce some genes. What type of genes I am going to introduce? The genes, gene is basically the DNA segment, no? Right? The genes in which both the strands, fine, the genes in which both the strands, right? The genes in which both the strands of the DNA, they will be transcribed. Just normally you know that the DNA strand having polarity 3' to 5', right? DNA strand having polarity 3', right? 3' to 5', only it gets transcribed now. This is basic. That's why bache, after molecular basis of inheritance I started this topic, because I want to make sure that you understand it. Okay, so this is the best way to revise it. Start the molecular basis of inheritance, then go for the biotechnology. So what am I saying? That in general, this is what we know when it comes to the transcription, right? From this double-stranded DNA, the DNA strand having polarity 3' to 5', it acts like a template. It is the one which will give us the mRNA. But now in RNA interference, we will be having a gene, the DNA segment, and this DNA segment, in this DNA segment, both the strands of the DNA, they will get transcribed. If both the strands they will get transcribed, so obviously you will get 2 mRNAs, isn't it? You will get 2 mRNAs, isn't it? Hanji Liharika, you are going to get 2 mRNAs, isn't it? And now if both the strands of DNA they are getting transcribed and you are getting 2 mRNAs, of course they are complementary to each other, right? It means they are complementary to each other. So if they are complementary to each other, right, if they are complementary to each other, so obviously they will form the bonds together and we will get a double-stranded RNA, right? We will get a double-stranded RNA. Clear bache? We will get a double-stranded RNA. Is that clear? Is that clear? So ultimately in RNA interference, we will be needing a double-stranded RNA that is, right, that is somewhere complementary to this target mRNA. I'll tell you how. Okay? So here I told you that externally we can culture, we can put that gene in that eukaryotic cell, plus sometimes bache transposons, you know transposons, yes. Do you know transposons? Transposons are the jumping genes which are present in the DNA. Sometimes that transposons are used to introduce that particular gene in that eukaryotic cell. Sometimes that viruses are used to introduce, right? Sometimes what? Sometimes the viruses are used to introduce that gene. So ultimately we have to introduce the gene within that eukaryotic cell so that we can get the double-stranded RNA. This point clear? This point clear? Now once you have this double-stranded RNA, here comes the role of dicer. What is dicer? Dicer is an endonucleases, right? Dicer is an endonucleases. What is dicer? Dicer is an endonucleases. It is going to cut the, right? It is going to cut the RNA within. So if it is, no doubt it is endonucleases, right? Leaving the terminal ends, it is going to cut the RNA. And this dicer na, it is RNAs, right? What is it? This dicer na, it is RNAs. What is the meaning of RNAs? That it digests the RNA, right? It is not DNAs. It is not DNAs. It is RNAs because it digests the RNA, right? It digests the RNA. So this dicer will come. So this dicer is going to cut this RNA, right? This dicer, it is going to cut this RNA, and you are going to get small, right? You are going to get small fragments, small double-stranded fragments. What are you going to get, Niharika? You are going to get small double-stranded fragments of this RNA. Now this is your siRNA. What is the meaning of siRNA? It is your small interfering RNA. It is your small interfering RNA. What is it, bache? It is your small interfering RNA. Clear, bache? Clear, bache? So now what is going to happen, right, bache? To the small interfering RNA, the risk will join. Basically, the proteins are going to join. This is risk. What is this risk? RISC. It is RNA induced silencing complex. It is RNA induced silencing complex. What's that? It is RNA induced silencing complex. Is that clear? Is that clear? What is it, bache? It is RNA induced silencing complex. It is RNA induced silencing complex. So basically these small interfering RNA, when these proteins will get attached, actually this protein is this risk, RNA induced silencing complex. It will get activated. Once it will get activated, right, now from this double stranded RNA you will have only single strand. Basically firstly what we did here, the double stranded RNA, with the help of dicer we are going to cleave it. We will get small double stranded RNA fragments. ठीक है बच्चे, these are small interfering RNAs, these are small interfering RNAs. Now what is going to happen to the small interfering RNA? You are having the protein, a protein will get attached there. It is a risk, right Krutika? It is a risk. It is RNA induced silencing complex. It is RNA induced silencing complex, right bache? So once it will get activated, right people, once it will get activated, what is going to happen? This is your protein complex. This double stranded RNA will become single stranded, and now because it is complementary to target mRNA, it will join with that target mRNA. It is going to join with that target mRNA. Okay? And once it will join to that target mRNA, students, once it will join to that target mRNA, it will cleave it, right? So now this target mRNA will not form protein, right? It will not form protein. Is that clear? This target mRNA, it will not form protein. ठीक है बच्चे? It will not form protein. Done बच्चे? Done. So this is in general RNA interference. Now in the case of pathogen, right, in the case of pathogen, what will be the story here in the case of pathogen? In the case of pathogen, the story is very simple, right? In my transgenic tobacco plant, I will be introducing nematode specific gene. Right, I will be introducing nematode specific gene. What is the meaning of nematode specific gene? Nematode specific genes means the genes, right? Jैसे in the nematode we have the target mRNA that we have to destroy. Right, in the nematode we know there is a gene that is forming a protein. That protein is pathogenic to my tobacco plant, so I have to destroy that protein, right? I have to destroy that protein. So the point here is bache, the point here is what we need to do, right? What we need to do, the point here is कि we have to, ठीक है, we have to, जैसे that particular gene in that nematode that is making the protein, and I don't want that gene to form the protein, right? So my transgenic tobacco plant is having the nematode specific gene. That gene will form double stranded RNA. That double stranded RNA na, it is, right, it will start RNA interference. You know the steps of RNA interference, and it will destroy the target mRNA of nematode. Because of that, nematode will not be able to infect my transgenic tobacco plant. That's how it works. Okay, that's how it works. This is what you need to remember. So write down the notes, bache, write down the flow chart, everyone. Right, so nematode specific gene, this is what we have to do for creating a transgenic tobacco plant. What will we do? Nematode specific genes. So how are we going to introduce that nematode specific genes? It is also a previous year question. How are we going to introduce that nematode specific genes? By using agrobacterium vector, right? That's why I told you about the agrobacterium vector, right? By using agrobacterium vector, right, these genes, they will be introduced into the host plant, right? So using using agrobacterium vector, right bache, this nematode specific gene, it will be introduced into tobacco plant. Fine. So nematode specific gene using agrobacterium vector introduced into tobacco plant. Then bache, sense and antisense, sense and antisense strand, sense strand and antisense strand RNA form, right? Basically both the strands of DNA, because now they are forming the RNA. So we have sense and the antisense strand, right? We have sense strand and the antisense strand RNA form, right? That is going to form, yes or no? That is going to form. So if you have sense and the antisense RNA strand, if you have sense and the antisense RNA strand, don't you think that it will join and it will form double-stranded RNA? That is how we are getting double-stranded RNA, yes or no? Don't you think the sense strand and the antisense strand, right, they will join, they will form double-stranded RNA? So after this, what is going to happen? The initiation of RNA interference will be there. After this, because once the double-stranded RNA is formed, there will be the initiation of RNAi, that is RNA interference. There will be the initiation of RNAi, that is RNA interference. Is that clear? There will be the initiation of RNAi, that is RNA interference, and after it, what is going to happen, bache? So this RNA interference will not let the target mRNA of the nematode to form, right, bache? Right, so silencing of specific, so it will result in silencing specific mRNA of nematode, right? It will result in silencing specific mRNA of nematode. Clear, bache? And finally, the nematode moves away, means nematode will not be able to infect that tobacco plant. This is how we will have a transgenic tobacco plant, right? Nematode moves away. Okay, so that is how, right? Finally, you can write down one more step. There will be, this is how the protection of transgenic tobacco plant will be there from the nematode. That's how it works. So nematode specific genes, with the help of agrobacterium vector, we will introduce that nematode specific gene into the tobacco plant. Now, because it is the nematode specific gene, they are introduced in such a way, right, that both the strands of the DNA, they will form RNAs. So now these RNAs, because they are complementary to each other, they will form double-stranded RNA. Once you have that double-stranded RNA, there will be the initiation of RNA interference. There will be the starting of RNAi, and after that RNAi, bache, there will be the silencing of that specific mRNA of nematode. Nematode will go away. That's how your transgenic plant, it will be protected. Clear, bache? That's how your transgenic plant, it will be protected. So this is about the Melodygyne incognitia part. This is about the yes, bache, RNA interference. Okay, do you know about flavor saver tomato? Anyone in the class, do you know about flavor saver tomato? That flavor saver tomato is also a transgenic plant, right? That flavor saver tomato is also a transgenic plant. Anyone in the class? Right, flavor saver tomato. So in the flavor saver tomato, because they have increased the shell life, you know, not tomato, their life is not too much, because when you talk about the hybrid tomatoes, their life is more. Why? Just a flavor saver tomato, in flavor saver tomato, right, basically what is there? The shelf life is more, the taste is superior, hai na? The shelf life is more, the taste is superior, because in flavor saver tomato, enzyme, this is important, enzyme polygalacturonase, right? The enzyme polygalacturonase is absent. What's the point there, bache? What's the point there? That when you talk about the flavor saver tomato, right, here in this flavor saver tomato, because its shelf life is more, it is superior in taste. Its shelf life is more, it is superior in taste, right? Its shelf life is more, it is superior in taste. Why? Because the enzyme polygalacturonase, it is absent. Enzyme polygalacturonase, it is absent. And do you know why that enzyme is absent? Right, basically enzyme absent means enzyme expression is not there, right? Enzyme's expression is not there, Leharika. Right, I am saying that enzyme polygalacturonase is absent, and obviously Samiksha is right. Because of that, there is delayed ripening. But the point is, how is it possible that a particular enzyme is not there? Obviously the RNA interference, right? RNA interference, we will not let this enzyme to express. We will not let that target mRNA to form that. Done? To form that. Right? Done, bache? Done? So all these examples are important and they are PYQs. Okay? So one another example is of Herudin formation. One another example is of Herudin formation. Do you know what is Herudin? Yes, bache. Do you know what is Herudin? Herudin. I hope you know about the Herudinaria. What is Herudinaria? Leech. What is that Herudinaria? Leech. Herudinaria is the scientific name or the zoological name of leech. So Herudinaria is having a chemical, that is Herudin, and this Herudin is nothing. It is the natural anti-blood coagulant. It is what? It is the natural anti-blood coagulant. Okay, it is the natural anti-blood coagulant. It is going to clot the blood. It will not let the blood clots to form. Okay, Herudin. Just we have heparin. So now what is going to happen, bache? Chemically synthesized. Right, basically we are not going to take that gene from Herudinaria, but yeah, chemically synthesized gene for Herudin formation will be introduced. Okay, so chemically synthesized gene for Herudin formation is introduced, right? It will be introduced in Brassica species, right? Brassica nippus. Okay, and now what is going to happen, bache? The seeds will have Herudin, right? The seeds will have Herudin. So Brassica nippus means we are talking about the rape seeds. So basically here the seeds are going to have the Herudin. So from these seeds the Herudin will be extracted. So that's how we can also produce this. So basically it is just like the molecular farming. It is the molecular farming. We are using the living organisms for getting the chemicals, right? We are using the living organisms for getting the chemicals. So these examples are important, bache. Okay, so examples are important. Okay, examples are important. Okay, so can you tell me about the golden rice? Let's discuss all the examples. Right, golden rice. So what is there in the golden rice? But golden rice is basically it is the food having better nutritional value, right? This is the example of biofortification. This is the word that I'm using again. Okay, bache, Snehit. Okay, so in which video you have commented? I'll check the video, don't worry. So yeah, biofortification, this is the word. Please do not forget this word. Biofortification, the process of increasing the nutritional value of the food. So now when you are talking about the golden rice, in the golden rice, right, in the golden rice, actually it's a source of vitamin A. It is a source of vitamin A. What is this golden rice? It is a source of vitamin A because it is having the precursor of vitamin A, that is your beta carotene. I'll tell you one very story. One very simple story. Do you know about rod cells? Do you know about rod cells? Just when we talk about the eye, now this topic eye is deleted from your syllabus, and trust me, I don't like it because I love this topic. You know, in your syllabus, eye and ear is not there. I'm not happy, I'm hurt. It should be there in your syllabus. It's very easy. So now when you talk about the eye, the innermost layer, it is having 3 layers now. So that innermost layer, you know, that is retina. So that retina is having photoreceptor cells. Yes, that retina is having what? The retina is having photoreceptor cells, right? PR cells, photoreceptor cells are there in retina, right? So basically that cells are your rod cells and the cone cells. So when we talk about the rod cells, right? C for color, C for cone, C for color. So in the bright light it will provide us the vision, and when it is the rod cell, in the dim light they are going to provide the vision. Right, in the dim light they are going to provide the vision, the rod cells, and in the dim light they are going to provide the vision. So these rod cells are having a pigment, that is rhodopsin. Which pigment is present in rod cells? That is rhodopsin. Okay, rhodopsin. So I hope you know rhodopsin. Opsin is the protein part here, and it is having the retinol aldehyde of vitamin A. right? Aldehyde of vitamin A. So basically for the formation of rhodopsin, vitamin A is required. What is required? Vitamin A is required. Yes people, what is required? Vitamin A is required. So if there is the, if there is the deficiency of, I hope now you can relate it, if there is the deficiency of vitamin A, it can result in night blindness. It can result in what? It can result in night blindness. Okay, so there are even countries where these vitamins, they are deficient. There are particular places. Okay, so now let's see golden rice. In the golden rice, what is done? In the golden rice, it is obviously genetically modified. It is genetically modified. It is having a precursor of vitamin A, right? It is having the beta carotene. That genes are introduced there. Okay, that genes are introduced there. They are having this precursor of vitamin A. That genes are introduced here. So now if a person having the deficiency of vitamin A, if they will start taking the golden rice, so obviously they'll be enriched with what? Vitamin A. Then all that issues, they will be resolved because that particular thing was deficient. That Hansika, that particular thing was deficient. So this is the example here. This is about the golden rice. Understood? This is about the golden rice, right bache? So we have another, that biofortified food. Just say there is one example of a potato. You will tell me this in the next class. It is having high protein content, right? It is having high protein content. So that is your homework bache. In the comment section you will let me know. In the comment section you people will let me know that you will tell me about the biofortified genetically modified crops. Okay, biofortified genetically modified crop, right? So this is about these genetically modified food. So you tell me, do you think that they are good, or any like, what do you think? Is it good to genetically modify the plants, the animals? Yes, what do you think? Is it good to genetically modify plants and animals? Anyone? Guys, please revert. Trust me, I'm not keeping well. I'm just taking your session because I really want to finish this chapter in this flow. So please be a support. Please revert. So what do you think? Is it good to make this genetically modified plants and animals? It's good to introduce such things. Is it good to introduce such things? 50-50. Like there are benefits also, and there are some harmful effects also. Disadvantages are also there. This is simple. See, it is not totally ethical. Why? Somewhere we are playing with the biodiversity. We are promoting certain genes only. Because of that, some other genes, they will be discarded. Isn't it? Some other genes, they will be discarded. That's the point. Secondly, my dear students, now let's say if we are having genetically modified crops, sometimes we can also be allergic to that. Some human beings, they are allergic to that, right? The cases for allergy, they are also being reported. Some human beings, they are allergic to that. Some cases of allergy, they are also being reported. And other than that, sometimes some antibiotic resistance, it can be like, let's say because we are dependent on that, so sometimes in our body antibiotic resistance is also there. Let's say now when we are sick, when it is a bacterial infection, we can take the antibiotic, right? Let's suppose if your body, it will become resistant to the antibiotic action, right? So if no antibiotic will act, okay, so how are we going to kill that bacteria? So issues like that can also occur. And as I said, it is not good when it comes to the biodiversity. We are playing with the biodiversity, right? We are playing with the biodiversity. So yeah, advantages are there. You are getting biofortified food, right? You can introduce your desired traits. You can have less post-harvest loss. There are many benefits also, but disadvantages are also there, right? Anything which is unnatural, it will result in disadvantages. Fine. So let's quickly read it. Let's quickly read this part, bache. So several nematodes parasitize a wide, right? Several nematodes parasitize a wide variety of plants and animals, including human beings. Okay. So nematode Meloidigyne incognitia, it infects the roots of the tobacco plant and it causes a great reduction in the yield. So a novel strategy was adopted to prevent this infestation, and it was based on the process of RNA interference. So here they are saying that it is this RNA interference is there in all the eukaryotic organism as a method of cellular defense. So be very specific. In all the eukaryotic organism, it's a method of cellular defense. Okay, bache? So this method involves silencing of a specific mRNA. Right. Here what we have to do, we have to silence a specific mRNA. How? Due to the complementary double-stranded RNA molecule. Due to the complementary double-stranded RNA molecule, and we have to prevent the translation of the target mRNA. That is what silencing is, right? That is what silencing is. Clear? That is what silencing is, right? So the source of that complementary RNA can be anything. You can use any virus to introduce that RNA, or mobile genetic elements, transposons can be introduced. So this part I have explained in the form of a flowchart, that by using agrobacterium vector we have to introduce nematode specific genes, okay, into the host plant, into the tobacco plant, right? So the DNA was introduced in such a way that both the strands will get transcribed and you will have sense and the antisense RNA in the host cell. So because two RNAs, they are complementary to each other, they will form double-stranded RNA that initiate RNA interference and thus silence the specific mRNA of the nematode. So that's how it works. You know the process in detail. Here you can see the diagram as well, right? So here you can see this is showing the roots of a typical control plants, right? And here transgenic plant roots 5 days after deliberate infection of nematode but protected through noble mechanism. Basically here deliberately, right, knowingly, forcefully, they are trying to infect the roots of this host plant with the nematode, but yeah, because it is transgenic, it was not able to infect it. Understood? So any doubt? Yes, bache, any doubt? Obviously PYQs are very important for biology. They are very important for biology. Okay, so you can see this double-stranded RNA, that is the dicer. So double-stranded RNA cleavage will be there with the help of that enzyme, right, bache? Then you will have small interfering RNAs, right, bache? You will have small interfering RNAs, right? And then with the help of that, risk formation will be there. RNA-induced silencing complex formation will be there. Okay, RNA-induced silencing complex formation will be there. Then finally that risk will get activated. Okay, so basically from the small interfering RNA, this is the double-stranded small interfering RNA, na, it will be cleaved, bache, and this is what you are going to get. Okay, so now this part, this siRNA with this protein, it will bind with the target mRNA, will destroy the target mRNA. Okay, will destroy the target mRNA. Ma'am, how the nematode transfer its gene to plant? It is not going to transfer its gene to plant. It is going to infect it with the help of a protein. But in our transgenic tobacco plant, we have already that mechanism that nematode will not be able to make its protein. And bache, if you know, not just related to this melodigyne incognitia, in general, if you know, जैसे Ascaris used to live, Ascaris is our endoparasite, है ना, Taenia solium is the parasite, right? So they can also live, right, within the host also. Fine. So biotechnology applications in medicine, so therapeutic use. Okay, that's what we need to discuss. What do we need to discuss? We need to discuss the therapeutic use. Okay, we need to discuss the therapeutic use. So RDNA technological processes, they have made immense impact in the area of healthcare by enabling the mass production of safe and more effective therapeutic drugs. By using this RDNA technology, we are making the better drugs. I'll give you the one example of genetically engineered insulin, right? I'll give you the example of what? Example of genetically engineered insulin. Okay, but before that you should know what exactly is the insulin. Bache, insulin is a protein hormone. What is insulin? It's a protein hormone. What is the meaning of a protein hormone? Obviously, it is about its nature. Like some hormones, they are steroidal in nature, some hormones they are amino acid derivative, some hormones, right, they are the protein hormones, the insulin. Now what is the point, right? What is the point here? See, you know, now second largest gland of our body is pancreas. Second largest gland of our body is pancreas. Yes, second largest gland of our body, it is what? It is pancreas, right? It is pancreas. Now in this pancreas, what's the point? You know, na, it's a composite gland. It's a composite gland. It is having the exocrine part that will make the digestive juices. It is having the endocrine part, right? The endocrine part. Do you remember this? So 99% is exocrine part. That 1% or 1 to 3% is endocrine part. So when we talk about the endocrine part of the pancreas, okay, so it is having the cells. You remember islets of Langerhans? Yes, do you remember that cells, islets of Langerhans? So that cells, right, like alpha cells, beta cells, delta cells, pancreatic polypeptide cells are also there. Alpha cells, beta cells, delta cells, and pancreatic polypeptide cells are also there, right? So among these cells, if you remember, the beta cells, they release what? They release insulin, right? They release insulin, which is a protein hormone. Now bache, whenever the blood glucose level is high, okay, whenever, even after meal, right, we are eating something that will be digested, then it will be absorbed, right, by our blood. In our blood, that glucose will be there na, then it will be transferred. So when blood glucose level is very high, so insulin will be released. This insulin will act on bache fat cells, that is your adipocytes, and it will act on your liver cells, that is your hepatocytes. And what will be done? That excess of glucose, yes bache, excess of glucose will be converted to glycogen, right bache? Excess of glucose will be converted to glycogen. Is that clear? Excess of glucose, it will be converted to glycogen. You know that if there is any deficiency of insulin, do you remember type 1, type 2, it results in basically diabetes, है ना? It results in basically diabetes. And you know that in India or all over the world, right, like all over the world, you know that people, they are affected with this disease, that is diabetes. Okay, that is diabetes. Okay, so scientists, they were like, let's form the genetically engineered insulin. Why? What is the need to form the genetically engineered insulin? Wait, I'll tell you. Come to this point. And we will also discuss we are also going to discuss the structure of insulin. Don't worry about that. So insulin used for diabetes was earlier extracted from the pancreas of slaughtered cattle and pigs. Okay, it was earlier extracted from pancreas of slaughtered cattle and pigs. Obviously it is unethical. From animals, we are killing them so that we can get the insulin from them, so that we can protect human beings. It is unethical, it is not good, and many individuals, it was the data is there, and as per that, many individuals, they get affected, they get allergies, they got allergies because of that insulin, right, because of that insulin. Okay, right? So insulin from an animal source though cause some patients to develop allergy or other types of reaction to that foreign protein. Okay, but when it comes to the insulin, okay, when it comes to the insulin, I told you bache that insulin is a protein hormone. What type of hormone it is? It's a protein hormone. So if it is a protein hormone, means it is made up of amino acid, right? If it is a protein hormone, it means it is made up of amino acid. It is made up of amino acid, yes or no? Yes or no? Obviously. So now this insulin, right, and especially I'll tell you the meaning here, especially the active insulin, it is having total 51 amino acids, right? It is having total 51 amino acids. Let me explain that. But insulin, it never forms it never forms as an activated hormone. No, insulin is secreted as proinsulin. What is proinsulin? It's a pro hormone. What is it? Like, do you remember the proteases? Renin, pepsin, prorenin, pepsinogen or propepsin. Do you remember that? That proteases, they are released in the inactive form. They are released in inactive form. So here same is the case. So when you talk about the insulin, it is secreted as proinsulin. Right? It is secreted as proinsulin. That is inactive form of insulin. Right? That is inactive form of insulin. Right, bache? It's proinsulin. It means it is inactive form of insulin. Right, bache? It is formed like this. So when you talk about this prohormone, right, it is having 3 chains of amino acid: A chain, B chain, or C chain. It is having 3 chains of amino acids. It is having 3 chains of amino acid. 21 amino acids in A chain, 30 amino acids in B chain, 31 or 34 amino acids. Right, I don't remember that exactly, or in some books 31 is given, in some books 34 is given. Right, so 31 or 34 amino acids in C chain. So basically when you are talking about the inactive form of insulin, right, I said na, insulin is always released in inactive form. So inactive form of insulin contains what? It contains A chain, B chain, C chain. A chain 21 amino acid, B chain 30 amino acid, C chain 31 or 34 amino acids. Clear bache? 31 or 34 amino acids. Right, 31 or 34 amino acids. Okay, so now let's discuss the structure here. It's something like this. Okay, it's something like this. Let me explain. This one is the A chain, this one is the B chain, and this one is the C chain of insulin. So A chain individually, I told you it is having 21 amino acid. So bache, how can you remember that? See, I think that 2, 3 will not be the problem. A, it's the first letter. So 1, 2, 1, 21. B structure is like this, no? Like use the structure, no? So that's how I remember. It is having 30 amino acid. It is having 30 amino acid, and that's mean. Okay, that is mean. So if you want to convert, right, let's say this is your inactive insulin. You want to convert that inactive insulin to the active one. You want to convert that inactive insulin to the active insulin. So in inactive insulin, there are how many chains? A, B, and C chain. So for converting it into the active insulin, we have to remove the C chain. We have to cleave the C chain, isn't it? We have to cleave the C chain. So ultimately, bache, in the active insulin, we just have A or the B chain. How many chains do we have? We have A or the B chain, right? A or the B chain. Is that clear? A or the B chain. Is that clear? So ultimately, what we need to remove? We need to remove the C chain. Okay? We need to remove the C chain. Like that will be the So see, this is going to be the active insulin. Fine. This is going to be the active insulin having A chain, having B chain. This is the active insulin. Okay, so how these 2 chains are connected to each other? How these 2 chains are connected to each other? This is another PYQ. Disulfide, with the help of disulfide linkage. Right, with the help of disulfide linkage. But please do not get confused here. See, these protein chains, they are made up of amino acid. So many amino acids they are going to join. So many amino acids they are going to join. So obviously they join with the help of peptide bond. Right, they join with the help of peptide bond. Isn't it? They are going to join with the help of peptide bond. But here we are seeing that how this A chain and B chain is attached. So it is attached with the help of disulfide linkage. With the help of disulfide linkage. Is that clear? It is attached with the help of disulfide linkage. Understood? Sure? Sure? So A, B and C. Okay, A, B and C, and A itself is having that sulfide linkage. Okay, A itself is having that sulfide linkage. It is also a previous year question. Basically intra chain sulfide linkage is also there. Okay, intra chain sulfide linkage is also there. So now from the structure one thing is clear. See, you know that when we need to make a, like, let's say now genetically by this genetic engineering, if we want to make the human insulin, okay, let's say if we are using the bacteria, of course the bacteria that we are going to use is E. coli. We will be using E. coli plasmid because we want the human gene to form the insulin. But do you think it's possible by using the bacteria? Plasmids are present in bacteria only. You know that you are not going to get it in eukaryotic cell. You know that eukaryotic cell are also not going to give you the restriction endonuclease. So one thing that we know is that in the case of bacteria, there is no RNA processing. In the case of bacteria, there is no RNA processing. Why, ma'am? What's the need of RNA processing? Here, this is the live example. Here, the protein that you are getting, it is in inactive form. We have to activate it. So obviously, we have to remove the part which is not giving us the coded information, which is not letting it to become the active insulin. Okay, so we have to remove the C chain. So we have A chain, we have B chain, we have the disulfide linkage which is attaching these two, right? Which is attaching these two. Now, what's the point here? So you know that in the case of bacteria, there is no RNA processing. In the case of bacteria, there is no RNA processing. In eukaryotic cell, there is RNA processing, but in the case of bacteria, there is no RNA processing. So if we are using E. coli, it's not possible to introduce the human gene here and to get the active insulin. No, if we will do this, we will get inactive insulin. It is of no use. It is of no use. So in 1983, an American company, hope you know this, right? In 1983, an American company, Eli Lilly, what they did, bache? Right, an American company, Eli Lilly, what they did? Yes, people. Yes, Umesh. What they did? They formed the A chain of human insulin and B chain separately. Right? They formed A chain and B chain of human insulin separately by using E. coli. By using E. coli plasmid. Okay, by using E. coli plasmid. Are you getting it? They form A chain and B chain separately by using E. coli plasmid. Okay? By using E. coli plasmid. So separately they synthesize. So because they synthesized it separately, right? So this A chain was exactly, it was exactly like human insulin. B chain was also like same. So then they joined it. Okay, then they joined it. Got it? Right? So that's how we got this genetically engineered insulin. It was brand named as Humulin, right? As Humulin. Okay, so this is about the use of this biotechnology or genetic engineering in the field of medicine. Okay, bache? Okay, so yes, you need to remember that A chain is having 21 amino acid, B chain is having 30 amino acid, right? They are linked together by disulfide bridges. Okay, so proinsulin is formed where extra C peptide is there. So we have to remove the C peptide and then insulin will become mature. Okay, so as I said, the American company 1983 Eli Lilly, an American company, they prepared 2 DNA sequences corresponding to A and B chains of human insulin. They introduced them in the plasmid of E. coli. They produced the insulin chains. Then A and B, it was extracted, it was combined, and the human insulin, it was formed. So that's the diagram. Fine, that's the diagram. Understood, bache? That is the diagram. Any doubt? Hanji, any doubt? Sure. Hanji, sure. So what? So how's the level of energy? How's the level of energy, people? No energy, ma'am. We are done, ma'am. Hanji. Is it clear? Okay, so next topic is the gene therapy, something very interesting, and it is very useful to treat the hereditary diseases, right? It is something which is very useful to treat the hereditary diseases. Hereditary diseases like what? Like your hemophilia. Okay, like hemophilia, like SCID, right? There are many hereditary diseases that can be treated with the help of this gene therapy. So what exactly is the gene therapy, bache? It's the collection of method. Gene therapy, ultimately we have to rectify, we have to replace a faulty gene with the normal one. We have to replace a faulty gene with the normal one. So gene therapy, it is an attempt to do this. It's a collection of method that allows correction of a gene defect that has been diagnosed in a child embryo. What is it? It's a collection of method. It's not a single method. It's a collection of method which will allow the correction of a gene defect, that has been diagnosed in the child or embryo. Right, that has been diagnosed in the child and embryo. So here genes are inserted into person, cell or tissue to treat a disease. So correction of genetic defect involves the delivery of a normal gene. So in simple words I told you, the faulty gene will be replaced by the correct one, right? The non-functional gene will be replaced by the correct one. Clear, bache? So this information is important, that first clinical gene therapy was given in 1990 to a 4-year-old girl with adenosine deaminase deficiency. Okay? With ADA. Okay. Chalo, let's talk about it. So the word is ADA, that is adenosine deaminase. Adenosine deaminase. But this enzyme is very important. Can you tell me the role of this enzyme? Yes, can you tell me the role of this enzyme, adenosine deaminase? Anyone in the class? Yes, Varun, Leharika, Umesha. ADA deficiency, you are right. Right, you are right. You are talking about the ADA deficiency. But what is ADA? What is ADA? Anyone? Form lymphocyte. So you are saying this enzyme form lymphocyte. But ADA is adenosine deaminase. You are right. You are right. And what do you think? This point is right, that it is present in the, right? You are right that it is present in the lymphocyte. But is it the only is it only present in lymphocytes? Yes. Is it only present in the lymphocytes? Tell me please. Is it only present in the lymphocyte? Tell me. But see, one student has mentioned it, that ADA is very important for purine metabolism, which is a fact. But here in what context we are reading this? See, ADA is very important for the development and maturation of the lymphocytes, right? It is very important for development and even for maturation of lymphocytes. Do you know what are lymphocytes? Yes, bache, do you know what are lymphocytes? Lymphocytes are the type of WBCs. Right, they are the type of WBCs, second most abundant type of WBCs they are. So tell me the first most abundant, tell me the most abundant WBCs. Tell me the name of most abundant WBCs. I said that lymphocytes are the second most abundant WBCs. What about the first one? Yes, what about the first one? Today in NEET Endgame batch, this is the chapter that we have started. This is the chapter that we have started. exactly neutrophils, their amount is maximum, and then there is the lymphocyte. So lymphocyte, for development and maturation of lymphocyte, we need the adenosine deaminase enzyme, and yes, it is very important for the purine metabolism. But now here we are reading it in association with the lymphocytes. So lymphocytes are the one that are going to fight with the infection, right? They are making the immune system. So this ADA enzyme, it is very important for our immune system. If there is no ADA, there will be no immune system. And imagine if we do not have the immune system, how are we going to survive? Right? How are we going to survive? That's the point here. Just in 90, like here, firstly, before starting anything, let's talk about the SCID. What is SCID? SCID is severe combined immunodeficiency syndrome. What is SCID, bache? It is severe combined immunodeficiency disorder, right? Or immunodeficiency syndrome. Severe combined immunodeficiency syndrome. Everyone write down the full form of SCID in the chat section, quick. Write down the full form of SCID in the chat section, quick. Severe combined immunodeficiency syndrome. Syndrome, if you remember, syndrome is what? Syndrome is a group of symptoms. Syndrome is a group of symptoms. Actually here it is the deficiency of immune system, right? SCID is a kind of primary immune deficiency disorder. It's a kind of primary immunodeficiency disorder. Now ma'am, what is the meaning of primary immunodeficiency syndrome? What I just said that SCID is the primary immunodeficiency syndrome. What is the meaning of primary immunodeficiency syndrome? Primary immunodeficiency syndrome means it is by birth. Okay, primary immunodeficiency syndrome is it is by Pavan Sai. Thank you so much. Right, I'm flattered. Thank you. Please now pay attention. So yeah, so SCID is severe combined immunodeficiency syndrome. It's the primary immunodeficiency syndrome because it is by birth. Okay, it is by birth. Now when you talk about the AIDS, acquired immunodeficiency syndrome, focus on the words acquired immunodeficiency syndrome, that we have acquired during the lifetime. Okay, that we have acquired during the lifetime. So that comes under the secondary immunodeficiency syndrome. Primary is by birth. So SCID is by birth. Okay, it's a kind of recessive disorder. It's very rare. But if any person is having the SCID, there the chances of survival are not there. If this SCID, right, if it is diagnosed in embryonic stages, okay, if it is diagnosed in embryonic stages, then bache, we can replace that faulty gene with the correct one. Then 100% cure will be there, because in embryonic stages, initially when all the blastomeres, they are just dividing, okay, they do not have their fate fixed. When all the blastomeres, they are just dividing and their fate is not fixed, right? When they are not differentiated, in that case, if we will recognize such genetic disorder, we can replace it and 100% cure will be there. But if you are recognizing it after birth, right, after the maturation, then 100% cure is not there. Why? Because we have to, in our body, in all the cells, right, we have to rectify that thing. That's the point. Okay, that's the point. Now I am saying that this gene therapy was first done in 1990 to a 4-year-old girl, right, having ADA deficiency. So this is very important for the immune system to function, right? This disorder can occur when there is the deletion of the gene of adenosine deaminase. So it is understood that if a person is having gene for adenosine deaminase, means the immune system will be functional, right? And if there is no gene, SCID can happen. So guys, first of all, be thankful for what you have. Right, be thankful for what you have. There are so many problems in this world. Okay, there are so many problems in this world, right? Be thankful for what you have. Okay, you are healthy, very good thing, right? No doubt sometimes we have that cold, fever, migraine, this and that type of issues, but it's still manageable. Imagine a person having SCID, the immune system is not functional, right? Even they cannot fight with the virus of cold. Okay, even a cold can affect that person very badly. There is no immunity, not even for any disease. Imagine this thing, right? So in some children, ADA deficiency can be cured by BMT, that is your bone marrow transplantation, right? By BMT, that is your bone marrow transplantation and enzyme replacement therapy. So basically ADA deficiency can be cured by two ways, Liharika. ADA deficiency can be cured by two ways. One is your bone marrow transplantation, that is BMT. Another is your enzyme replacement therapy. Both are not 100% efficient, right? So basically here what we have done, functional ADA is given to the patient by injection, but the problem with both of these approaches that they are not completely curative, right? It's not 100%, right? It is not giving the 100% cure. Okay? So what we have to do as a first step of genetic engineering, right? Let's say our Chintu is having ADA, right? Let's say our Chintu is having ADA deficiency. So what will be done, bache? What will be done from Chintu's body? Lymphocytes will be taken. From its blood, lymphocytes will be taken. So outside the body in lab, right, they will be cultured. What will be done, bache? They will be cultured. What will be done, people? They will be cultured. Why are we culturing it? Because we know that ADA is deficient here. So what if in these lymphocytes, if we will add complementary DNA for ADA? What if we add complementary DNA for ADA? Now ma'am, what is the meaning of this thing here? See bache, we know that this condition is because of the absence of, right? We know that this condition is because of the absence of what? Yes, bache, because of the absence of what? Because of the absence of ADA. So what will be done? First of all, we will take the gene which forms the ADA. Now bache, what will be done? First of all, we will find out that the gene that will form ADA, adenosine deaminase. Now we will take that gene, right? The E. coli plasmids will be used. I know you don't know about the E. coli, so please pay attention here. E. coli plasmid will be used, and this gene, like now let's say, if in the plasmid, if I will introduce this gene is going to form the mRNA there, then protein. It is going to express itself. So basically what will we do? We will pick up that mRNA for that ADA, and that mRNA, basically that mRNAs, right, it will be inserted in a retrovirus. Ultimately, bache, the point is, let me tell you, right, let me start it with that NCERT flow. So we are going to take out the lymphocyte of a patient, we will culture it, and with the help of retroviral vectors, right, with the help of retroviral vectors. What is the meaning of retroviral vectors? What are retroviruses? What are retroviruses? Retroviruses are the one, right? Retroviruses are the one where RNA is acting like RNA acts as a genetic material, right? These are the viruses where RNA acts as a genetic material. This is what retrovirus is, right? This is what a retrovirus is. RNA is acting like a genetic material. So now if I'm going to use retroviral vectors, so do you remember that point? That we have to make them harmless, right? We have to make them harmless. It's just that we are using their natural ability. Retroviruses, they have the tendency that they can introduce their genetic material into the host cell. They convert normal cell into the cancerous one. This is what you know, Veshu. They convert the normal cell into the cancerous one. This is their natural ability. So what if we remove the harmful gene and we introduce the useful one? Again, this vector is going to behave like the same way. Again, this vector will introduce its genetic material into the host cell, right? But now it is not passing that pathogenic gene. It is not passing that pathogenic genetic material, right? The infectious genetic material, but it is passing our useful genes. That is what we have to do. So we will take the retroviral vector. Obviously, it is harmless. That is it's adenovirus. Actually, it's adenovirus that will be used here. Now, in this virus, we have introduced the RNA for the formation of complementary DNA of that ADA. How can we do that? Right? We can take the E. coli's plasmid. In that E. coli's plasmid, we have already introduced the gene for ADA. Now from that E. coli, right, they both, E. coli and retroviral, they both will be infected. So ultimately moral of the story is that we have to pass, we have to pass that particular, we have to pass that cDNA, we have to introduce here that RNA, right? We have to introduce that RNA here in this retroviral vector. We have to introduce that RNA here to that retroviral vector. Are you getting it? Are you getting it? So a functional ADA complementary DNA with the help of retroviral vector, we will introduce it into the lymphocyte. So how can we do that? Firstly, we have to introduce within this retrovirus, we have to introduce that RNA for the formation, right? For the reverse transcription of the ADA. Is it clear or not? Is it clear or not? Tell me. If it is not clear, I'll explain it again. Please let me know. Please let me know. Is it clear or not? Okay, Veshu, the point is, see, it's a retroviral vector that we are going to use here in this gene therapy. This viral vector is having the RNA because it is a retrovirus, its genetic material is RNA. Now, you know that it is the natural tendency of the viral vectors that they use the host cell's machinery for making their multiple copies. Now in this retroviral vector, we have already introduced the mRNA that can, with reverse transcription, you know na reverse transcription, जैसे you have mRNA with the help of reverse transcriptase enzyme, right? With the help of reverse transcriptase enzyme from this mRNA, we can form single-stranded complementary DNA. Actually, when mRNA is used to form the DNA, that DNA is also what? Single-stranded na? We call it as complementary DNA. Which enzyme is going to help there? The reverse transcriptase. Which enzyme is going to help there? Reverse transcriptase. And slowly from this complementary DNA, the double-stranded DNA is formed. This is the procedure actually, right? This is the procedure actually. So in my retroviral vector, right, in my retroviral vector, if that retroviral vector is having the mRNA which can form the ADA, that is going to be helpful. That is going to be helpful, right? We can use this vector to put this mRNA into our lymphocytes. The lymphocytes which are cultured outside the body, the lymphocytes which are taken from patient's blood, which are cultured outside the body, within these lymphocytes, with the help of retroviral vector, we will introduce that complementary DNA because this virus is having the reverse transcriptase. It can convert its RNA into the DNA because it wants to introduce its DNA into the host's DNA so that it can use its machinery. So that's how we will pass it, right? That's how we will pass it. Now is it clear? Now is it clear? And if someone asks you that how are you going to introduce that mRNA in retroviral vector, that we will done with the help of E. coli, right Sabha? That will be done with the help of E. coli. Okay? So now these lymphocytes bache, right? Now these lymphocytes, what will be done to these lymphocytes? So see, as a first step towards the genetic therapy, gene therapy, sorry, lymphocytes from the blood of the patient, they are grown in a culture outside the body. So functional adenosine deaminase enzyme, complementary DNA, by using a retroviral vector, it will be introduced into these lymphocytes, which are subsequently returned to the patient. So however, these cells are not immortal, right? These cells are not immortal means they are not going to live always. They are mortal means they will die. Immortal is someone who's not going to die ever. Who's not going to die ever. Okay, so these lymphocytes are not immortal. For the time being, that patient will have the immune system. Why? Because ADA is introduced in the lymphocytes. But that lymphocytes will also destroy. Then again we have to do this procedure. Again we have to take the lymphocytes from the blood of that particular patient. Again we have to culture it. Again we have to introduce that ADA there. Clear, bache? Clear, bache? So that is how it works. So it is not a permanent cure if it is not. See, it is not a permanent. It could be permanent if we will, figure out that thing in the embryonic stage and we replace the gene. Otherwise, it is not permanent. Okay? Okay? Right? So next is the molecular diagnosis, then transgenic animals, then ethical issues. So that's what we need to discuss and the chapter will be over. So how's the energy? How's the Josh, guys? Let's finish it in next 30 minutes. Let's finish it in next 30 minutes. What say? Sure. Okay. Okay, so now what is molecular diagnosis? See, if we need to treat a disease, firstly we should know about that disease. है ना, we should know about that disease. जैसे, see, we go for the blood testing and all. These are the conventional ways, old ways, right? जैसे when there is the examination, blood test is there, right? When our blood is examined, what? We used to know about the infections. If total leukocyte count is there, if total leukocyte count is more, this is what doctors used to say, na, that infection is there in body. It's like that, right? It's like that. But when are we going to see the results in the blood test? When are we going to see the results in the urine test? But when that particular pathogen has acquired our body, right? When that particular pathogen has started affecting our body, then we will, by these blood tests, by these urine tests, then we can see the results, right? What if we want to diagnose a disease little early? If we will diagnose a disease early, we can cure it, right? We can cure it. That's the point here, right? That is the point here. See, using conventional methods of diagnosis like serum and urine analysis, early detection is not possible. Early detection of that particular pathogen in our body, of the, if there is any, see, what can be pathogenic? Let's say if there is a particular pathogen, it's not mandatory that entire pathogen will be pathogenic, right? It is not mandatory that that complete organism, right, Sanjay, that complete organism will be pathogenic. Of course not, है ना? Of course not, right? Maybe in that cell, maybe there are some proteins which are pathogenic, which are generating the immune response. It can be anything. So basically, even if in our body, if there is any foreign substance, even if there is a very minute, very small fragment of protein, even if it is there is a very small polysaccharide, right, we can diagnose that, right? We can detect that, but with the help of these molecular diagnosis method, not with the help of that conventional ways like the blood test and that, what is that urine test and all. Okay, urine test and all. And these techniques, जैसे rDNA technologies, PCR, remember PCR, denaturation and healing extension. In the last class, that's what we have discussed, na Sanjay? Right? If you are new to our channel, guys, quickly hit the like button, and I want to see maximum people here in this course. That is your NEET end game. The link is there in the description box. Just click on the link there and be the part of this batch. It is the ultimate crash course where there will be the proper revision, quality content will be provided, proper sessions and tests will be there, right? Right, proper session and tests will be there, proper practice will be there in the lectures. So be the part of that batch, okay? So now come back to this topic. So rDNA technology, right? Polymerase chain reaction. Remember PCR, DAE. 3 steps are there in PCR: denaturation and linear extension. It is used for amplification of gene. And then bache, enzyme linked immunosorbent assay, ELISA. Enzyme linked immunosorbent assay, ELISA, right? Enzyme linked immunosorbent assay, ELISA. Is that clear? Is that clear? So here in this ELISA, basically it is on the basis of antigen antibody interaction, right? Its base is antigen antibody interaction. So all that things, right, they are the molecular diagnosis by which we can detect that pathogen, any protein, any polysaccharide of that pathogen very early. Okay? Right? So see, it is written here that presence of pathogen is normally suspected only when the pathogen has produced a disease symptom. But by this time, the concentration of pathogen is already high in our body, right? Already high in our body. So very low concentration of bacteria or virus at the time when symptoms of the diseases are not yet visible, it can be detected by amplify the nucleic acid by PCR. Okay? So you know that PCR is routinely used to detect HIV in suspected AIDS patient. So please remember, in AIDS, no doubt, right? right? In AIDS, no doubt PCR is used, right? But confirmatory test is, this is a previous year question, so please mark it. But confirmatory test is Western blotting. Is it Western blotting or something else? Do you remember that? Confirmatory test in the case of AIDS is, in the case of HIV is, right? It is the Western blotting, but yeah, PCR is also used. Okay, PCR is also used, right? PCR is also used. Clear, bache? Right? So it is also, this PCR is also used to detect the mutations in the genes in suspected cancer patients too. So it's a powerful technique to identify many other disorders. That's what you need to remember. Okay, that's what you need to remember. So please quickly recall the PCR. So which enzyme is used in PCR? Which enzyme is used in PCR? Which enzyme, Krutika, which enzyme is used in PCR? Be quick. Be quick. Which enzyme is used in PCR? Taq polymerase. Okay, that is Taq polymerase. It is obtained from Thermus aquaticus. Now, bache, here they are talking about the probe. Probe is something which is used to detect, right? Right. Probe is something which is used to detect. Jaise DNA probe ya RNA probe. We can take a small segment of DNA that will be radioactive. Okay, we can use that probe to identify if there is any mutation in the DNA or not. Jaise here, see, a single stranded DNA or RNA which is tagged with radioactive molecule is allowed to hybridize to its complementary DNA in a clone cell, followed by detection using autoradiography. Very simple. Let's say if there is any doubt of mutation in our body, let's say there is any disorder or there is any chance of disorder, jaise PCR can amplify the gene, right? PCR can amplify the gene. Am I right? PCR can amplify the gene. When that gene is amplified, I told you that we can use PCR to detect mutation. So obviously the gene is amplified. You have that particular gene, you know the sequences, right? And let's say to that gene, you are making it single stranded, right? You are separating that DNA fragment. Let's take an example. Now you know that ideally that sequence, the DNA segment should have a proper sequence, right? Whatever sequence it can be, that particular DNA should have a proper sequence. But because you are testing for mutation, so complementary to it, you will take a DNA probe, right? What is special about this DNA probe? That it is radioactive. So obviously you will try to hybridize it. If it will get hybridized, because it is complementary to it, if it will get hybridized, no mutation. But if it will not get hybridized, it's a sign that you were expecting a particular sequence, it was not there, means mutations, right? It means mutations. Clear, bache? Clear, bache? So the clone having the mutated gene will hence not appear on photographic film. So if it is not mutated, they both will join, right? They both will join, and by using autoradiography, remember gel electrophoresis, later on that gel is exposed to the UV rays, we will see the bands, right? So later on, that is autoradiography. So if on autoradiography, if you will see that, if you will detect, then that's fine. If this DNA probe, if it will not get attached here, so it's a clear-cut sign that mutations are there, right? That you were expecting a sequence, but that sequence was not there. Okay? So then comes the ELISA, enzyme-linked immunosorbent assay, which is based on the principle of antigen-antibody interaction. So antigen, which will generate immune response, the antibody, the clumping will be there, the agglutination will be there, right? That's our base. So basically by ELISA, the infection by pathogen can be detected by presence of antigens or by detecting antibody synthesized by the pathogen. It's very simple. Just let's say we have Chintu, our Chintu is not keeping well. Okay? We have Chintu, our Chintu is not keeping well. In the Chintu's body, I want to check that is there any pathogen or not? Is there any pathogen or not? There are two ways. Firstly, I can even, let's say, let's not consider Chintu is well or not. Like we use these tests for the early diagnosis. So let's say in Chintu's body, few symptoms are there. We want to check whether a particular pathogen is there or not. So what can we do? Now we can even test for antigen, right? If a doctor is having a prognosis कि this can happen, that can happen, ठीक है, so accordingly a test will be performed, a particular antigen can be detected obviously. Or there is another approach also. Let's say if you are not focused on the antigen part, if let's say if there is any antigen in my body, if there is any foreign substance in my body, so obviously my body's immune response is going to make the antibodies against it. If there is any Nandini, if there is any foreign substance in my body, obviously my body's immune system is going to make antibodies against it. So I can either detect that antibody. If I am a doctor, I am suspecting that this can happen, so I will recommend that test na ki this should be checked. It's like that, it's like that. So that's why here in this NCERT line it is written कि we can even detect the presence of antigens, or by detecting the antibodies synthesized against that pathogen. Then my body, even if that antigen is in less amount, Hansika, my body is going to react against it. My body will form the antibodies, and we can also work on that. So there are different ways of ELISA as well. The direct ELISA, basically बच्चे, a plate will be there, the wells are there in that plate, so we are going to put the body, right, we are going to put that body fluids there, then the antigen will be immobilized, right, we will make sure that antigen is immobilized, it is not moving. Then complementary to it, if we are suspecting any antigen, complementary to it, antibody will be added, and to that antibody, can you see this, it is attached. What is it? What is it? Anyone in the class? To this antibody something is attached. What is it? Anyone in the class? What is it? Tell me quickly. What is it? Hanji, what is it? No, it is the, देखो, very simple it is. जैसे let's say you are taking the body fluid, right, you are taking the body fluid, you are adding the body fluid here. We will make sure, let's say if we are checking that antigen, we want to confirm any antigen, so here na we will make our antigen immobile, it should not be movable, it should not be movable. Let's say it is fixed here, right, immobile, it should not be movable. So now we are going to add some antibodies, right? So if whatever we are suspecting, if it is there in the blood, so obviously that antibodies should bind to this antigen. Antigen, antibody, you know na, they show the clumping, they should bind here, okay? So we will again do the washing, the antibodies which are not bounded, they'll be washed. And now what will I do? See, there are different ways. Just in direct ELISA, what they have done, the antibody that they have attached na, the antibody that they have added na, they have attached enzyme to it. Okay, so now I can add the substrate for that enzyme. If it will show the color, means antigen is present. It's like that. Okay, so I'm not discussing it in very much detail. So indirect ELISA mein, the antigen, immobilized antigen, antibody will join, right? So here we also add another antibody, secondary antibody, which is complementary to this antibody, and enzyme is attached. So it's like that. So basically here we check the color and the intensity of that color, right? We check the color and the intensity. Accordingly, we relate it with the intensity of that pathogen in our body. Okay? So as of now, what will I suggest? That only focus on this part. Okay? So enzyme linked immunoassay, it is based on the antigen antibody interaction. So clumping will be there. So color production will be there. Ultimately that color production that we need to focus. Okay? Okay? So next bache is transgenic animals. They have given the examples here. Right? We are about to finish it. We are about to finish it. So guys, show some energy. Let's finish it till 11. Everyone, let's finish it till 11. So जैसे you know ना, transgenic animals में cow, sheep, goat, right, they give us the therapeutic human proteins in their milk. There are different examples here. So transgenic animals are the one, they are having the foreign gene, extra genes, जैसे rats, rabbits, pigs, sheep, cow, fish, right? And 95% of all the existing transgenic animals, they are mice. How can we use it? जैसे let's say if you have the transgenic mice, okay, now in that mice you can introduce the gene. Just let's say I am suspecting that mutation in a particular gene is causing a XYZ disorder. I'll give you one example. Just I am suspecting that there is a gene A, and in the gene A, if there is the mutation, it is causing the disorder X. Okay? There is a gene A, and if this gene A, if it will get mutated, it will result in a disorder. The name of the disorder is X, but I want to check it, I want to make sure it. How can I? How can I? It cannot be the hypothesis. I should have that result. No, I cannot focus on that theory, right? No one is going to believe, right? No one is going to believe what I'm going to say, right? They need the experimental proof now. So let's say this is what I'm suspecting. So I will make a transgenic mice. I will make a mice, right? In that mice I will add this mutated gene, right? So even after adding it, if I'll see that my mice is suffering with the X disorder, it's a kind of confirmation. So basically these transgenic animals, they are used to study any disorder, right? To see the result of mutation, to see the result of absence and the presence of gene, any gene which is present in excess, if any gene that is absent, right? We can use it to understand. We can use these transgenic animals to understand the physiological processes as well, right? To understand what? To understand the physiological processes as well, that how in our system that particular, you know, that particular cycle is going on. We can also use these transgenic animals to study a particular disease, the life cycle of a pathogen, because if we will study the life cycle of a pathogen, we'll get to know about the, right, we'll get to know about the spaces, about the points where, you know, we can make the disease, make the drug, okay, this is the step that we can stop. Okay, so that's how these transgenic animals, they help. So here, right, from your examination point of view, what is important? Examples. Just transgenic animals can be specifically designed to allow the study of how genes are regulated, right, how they affect the normal function. Just a study of complex factors involved in growth, such as insulin like growth factor. How are they helping in growth? That is what we can study by introducing genes from other species that alter the formation of these factors. So that's how we can use them. Secondly, for the study of disease, I already told you we have many model organisms, right, for studying the disease like cancer, cystic fibrosis. What is cystic fibrosis? What is cystic fibrosis? What is cystic fibrosis? It affects lung. Acha. Desiti, have you seen the movie? There is one movie, na? Hi Papa or Hi Nanny, Nana, something like that. Is it on the basis of that, or you actually know cystic fibrosis? Right? It is even in the genetics. Yes, bache, it is even in the genetics. Hi Nana. See, hi Nana. I told you. Hi Papa, hi Nana. है ना? अरे yeah, cystic fibrosis is even there in the genetics. It is even there in the genetics, cystic fibrosis. So do check it. What is it? Is it autosomal recessive, autosomal dominant? What is it? Please check it. Please check it. Okay, cystic fibrosis. So there is no doubt lungs are affected, but what is happening actually? What is happening? It is somewhere related to the ion transport. So which ion? So that's your homework. That's your homework. Okay, I want to see the answers in the comment section. And if you are actually serious for your NEET examination, please do this thing, right? Please start solving these questions. Okay. Okay, so for studying the RA, rheumatoid arthritis, just I hope you know that it is autoimmune disorder. Rheumatoid arthritis is autoimmune disorder, right? Right, then Alzheimer disease. So for studying of such disease, right, we have the model organism, and for the biological products, right? So here this part is important. So medicines required to treat certain human disease, they can contain biological products, but such products are very expensive. So transgenic animals that produce useful biological products can be created by introducing that particular gene. Just here they are giving us the example of alpha-1 antitrypsin. Very important it is. Bache, do you know about the disease emphysema? Do you know about emphysema? Emphysema. It is because of the continuous cigarette smoking, right? The lungs will be damaged. Emphysema. Right, the chain smokers, they get this disease, emphysema. So for treating emphysema, alpha-1 antitrypsin is required. So if I don't know anything about it, right, if I don't know anything about it, emphysema is the word. I know it is because of the cigarette smoking. The lungs, the alveoli, they will lose their elasticity, right? That fibrous kind of growth will be there. The gaseous exchange will be affected. Okay? This is what we already know from the human physiology. So alpha-1 antitrypsin is used to treat emphysema. It is antitrypsin. What is trypsin? Niharika, what is trypsin? Trypsin is a protein digesting enzyme. trypsin related with the digestion and absorption, na, trypsin, it is in pancreatic juices. At least this much you remember, na, trypsin is a protease. It is going to digest the protein. So if we are using antitrypsin to treat emphysema, matlab, what it means? It means that in emphysema trypsin is present, right? Trypsin, and we have to stop its function. So alpha 1 antitrypsin is given. Alpha 1 antitrypsin is given. So start relating the things, right? Start relating the things. Okay? So similar attempts are being made for the treatment of phenylketonuria. Hope you remember it is autosomal recessive. So bache, the name of the diseases which are given in your NCERT, not just in your chapter principles of inheritance, even in your biotech, even in your human health, wherever the diseases are written, na, please write it down on a paper and please check that you know about it or not. Okay? That you know about it or not. So phenylketonuria and cystic fibrosis. In 1997, the first transgenic cow, Rosie, right? It was the first transgenic cow, Rosie. In 1997, first transgenic cow, Rosie, was produced, right? And it was enriched with milk, right? So that milk was proteinaceous. Okay, so 2.4 grams per liter. See, it produced human protein enriched milk, right? So the milk contained the human alpha 1 lacta, human alpha lactalbumin. Another important MCQ. Take care, please mark it, right? So it is very much nutritious, even more nutritious than the mother's milk. Okay, mother's milk. Then vaccine safety. For checking the vaccine safety, we use the transgenic animal. For checking the chemical safety also, the transgenic animals are used. Understood? So next is bache ethical issues. So before that break I was asking you, what is GEC, Genetic Engineering Approval Committee. देखो बच्चे, genetic engineering is good, but if it is used for the benefit of mankind, otherwise it is very dangerous. No doubt we are playing with the diversity, and there are other factors also. They say, let's say if there will be no check on such researches, many scientists, out of their curiosity, they can do anything. They can try to make any organism, any modified organism. They can try to do anything, like let's say they can even make the bioweapons. Okay, they can even experiment on the human beings. So obviously there should be a committee that is going to set the rules, that will check that the research, that what are they actually doing, and their research, is it benefiting the mankind or is it harmful. So all that factors should be considered. Okay, all that factors should be considered. So here what is the point? That is GEAC, Genetic Engineering Approval Committee, right? So which will make decisions regarding the validity of GM research and the safety of introducing genetically modified organisms for the public services. So this is what they are going to do. Okay, this is what they are going to do. ठीक है बच्चे, ठीक है बच्चे, GEAC. So ethical issues are there because the manipulation of living organism by human race cannot go on any further without regulation. So some ethical standards are required to evaluate all that thing. So here the question will come from GEAC. That's what you need to remember. Done? That's what you need to remember. So next thing is bache, it is related to the biopiracy and the biopatent. Right. Firstly, I'll tell you about the biopatent. Bache, see, whenever there is any innovation, whenever there is any nobility, non-obviousness, right? See, focus on these things, focus on these words. Yes, focus on these things, please. So biopatent, let's talk about the biopatent first. Let's say if there is any innovation, right, like nobility is there, non-obviousness is there, and obviously the utility. These are the things that we consider when we talk about the patents. Patents, they are the special rights, right? Patents, they are the special rights. Okay, Padam Mishra bache, on this channel I have posted one video related to AFMC. Okay, you can check this video, you will get the details of AFMC. Okay, you want to join the AFMC, you will get an idea that how you have to prepare. Oh, Babaji is here. Hi HSP sir. Sir, even I am missing your classes. You are not posting any video and we are not posting any short. What's going on, sir? What's going on, HSP sir? We are upset. You are not posting any shot. We are waiting for your tricks. है ना, we are waiting for your tricks. So promise us that you are going to post it as soon as possible. Huh? Chalo. So now come to this point. So patent means any special rights, right? Any special rights which are given to a human being or an organization, right? If they have discovered something, right? Who's going to provide it? It depends upon that innovation, the state government, the central government. It's like that. The special rights, right? The special rights will be provided to the person who discovered that. Okay? So without their basically it is just like a copyright issue. Without their permission, we cannot commercialize that particular product. Okay? So when it is a biopatent, obviously you are talking about the living organism. So on what basis we are going to decide that this is a new innovation, this is something new? It can be anything. It can be any organism that they have reported. It can be any protein, its benefits, yeah, any enzyme. It can be any technique, right? If they have given any technique, so obviously what will they get? What will they get? They will get the right, they will get the special rights on it. So whenever you talk about that type of innovation, now this is what we consider: the novelty, non-obviousness, and the utility. First of all, that should be beneficial for the human being, that should be beneficial for the environment. That's the first thing. Then it should be new, right? It should not be like that someone has already explained it or anticipated it. If let's say there is any research that has anticipated that okay, that can happen, still that will not be considered as a innovation, right? So it should be new, totally new, and obviously non-obviousness, like no one has ever even hypothesized it. Okay, this can happen. It's like that. Then that person will get the rights for that discovery. So this is the biopatent. Okay, this is the biopatent. Clear bache? Clear bache? Yes, this is about the biopatent. So official right from a government granting an inventor or establishment sole monopoly to the use of that invention. So see, this is written here. So see, the modification usage of living organism for public service has also created problem with patents granted for the same. So there is growing public anger that certain companies are being granted patents for products and technology that make use of genetic material plants. Basically here they are talking about the biopiracy. So first of all, tell me, biopatent clear? What is the biopatent? Is it clear? Is it clear? Now what is biopiracy? Bio, what is there, means we are talking about the living organism, any animal, any plant. Piracy, right? To steal something, right? Or to use something without the knowledge of that particular discoverer. It can also be like this. And it is not just related to that patent part, bache. Just let's say our country is having some resources. Okay, we are not claiming it. But some other country is using it without our knowledge. That is also the piracy, right? If let's say something belongs to our country, right? But some other countries, they are getting the patent right for that. It is also the biopiracy. It is also the biopiracy, and strict rules should be there. Just a best example is of that basmati rice. Even not just the basmati rice, bache, not just the basmati rice, even for the neem, even for the use of turmeric in wound healing, right? You know that in our country we have been using such things from so many years, right? So many years. But other countries, they are getting the patent rights for it. Why? Because we are not aware of it, right? They say the University of Mississippi, they got the patent for the use of turmeric in the wound healing, but in our country we have been using it from so many years. So that all things comes under the basmati rice. Just I'll give you one example here, right? I'll give you one example here. Rice is a very important food grain. Okay, it is a very important food grain, the presence of which goes back thousands of years in Asia's agricultural history. So there are an estimated 2 lakh varieties of rice in India alone, right? So diversity of rice in India is one of the richest in the world, and basmati rice is distinct, you know that, for its aroma and flavor. And 27 documented varieties of basmati, they are grown in India. Okay, they are grown in India, and their reference is also there in the Asian textbooks, right? But in 1997, an American company, they got patent rights on basmati rice through the US Patent and Trademark Office. So an American company, they got the patent rights for that basmati, right? This allowed the company to sell a new variety of basmati in US and abroad. So this new variety was actually bache the hybrid, kind of hybrid, right? It was derived from Indian farmers variety, Indian basmati. They were crossed with semi-dwarf variety and they claimed it as their innovation. So this is what? This is biopiracy. This is piracy, right? So patent extends to functional equivalents, right, implying that other people selling, so basmati rice could be restricted by the patent, right bache? Right bache? Same was the case with the turmeric and neem. Jaise turmeric mein I told you in 1995 the US patent office, they granted the patent to the University of Mississippi, the use of turmeric in wound healing, right? And even the neem bache. So in 1996 Vandana Shiva challenged the patent, right? Vandana Shiva challenged the patent given to that neem, right? That neem, you know na, it is it kills the fungus, right? That neem oil can be used to kill the fungus. So some other country, they got the patent right for that. Okay, they got the patent right for that. So Vandana Shiva challenged the patent granted for this use of neem oil. Okay. And for the turmeric, Dr. R.A. Mashelkar, okay, for turmeric, Dr. R.A. Mashelkar, he challenged that patent. So the awareness should be there and there should be strict rules, okay, there should be strict rules. So this is basically the biopiracy, right? It is used to refer to the use of bioresources by multinational companies and other organizations without proper authorization. So I really don't think that I need to explain it. You can read it by your own. And yes, this paragraph is also very important, that Indian Parliament has recently cleared the second amendment of the Indian patent bills that takes such issues into consideration, including patent terms, emergency provision, research and development initiative. That's it. That's all. One more chapter down. Okay, so this is more than sufficient. This video is more than sufficient for biotech applications, even for that biotechnology principles and processes. So bache, human reproduction, reproductive health, even sexual reproduction in flowering plants, even your evolution, principles, molecular, this biotechnology, both the chapters. And now on 23rd of Jan, we are going to finish human health and disease, right? So kind of your 12th syllabus is over. Okay, so we'll see, maybe I'll take microbes in human welfare, or your Pankhudi ma'am, I'll discuss that with her. Okay, you will see it on our channel, and then we'll move to the class 11 syllabus. So from my end, I have finished the class 12th part. Only one chapter is there. Okay, right, only one chapter is there, the zoology part I'm talking about. So now it's all up to you. Still you have time, still you can manage, right bache? Please be sincere, please follow a proper strategy. If you are not able to focus, be the part of our batch. Okay, be the part of our batch if you're not able to focus, if you're not able to study by your own, right? YouTube is a public platform. We are giving you content free of cost, but still, right, there are many distractions on this platform. If you are, you know, dedicated, if you are not getting distracted, then okay, fine, you can use these classes. But bache, if you actually need that decorum na, that where you are studying, you are competing with other NEET aspirants, okay, you have to answer a question, there is a test, if you need that, and right now you actually need that, okay, so this batch is for you, this batch is for you. Okay, so I'll share the PDF in the Telegram group. Stay tuned. So see you all on 23rd January, and I want the answer of all the questions in the comment section today. If you're a serious NEET aspirants, this is what I want, right? This is what I want in the comment section. Okay, so take care, bye bye. Thank you so much.